Hi, Olga.
Hello, hello, hello. We are starting in about a minute. So people are coming in already, and we are broadcasting on a YouTube live. So just give me another minute and we're going ahead and we will get started. Thank you very much for joining us. Okay, I think we are ready and we can go ahead and get started. Welcome back, everybody, to day 4 of May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. We are very excited to see so many of you coming back. It means that we are not completely exhausted yet. And this is good because we have more really interesting things to talk about this week and also the following week. So if you happen to join us today for the first time, let me just go over some logistics to remind you. So at the bottom of the screen, you have the chat box. So please use this chat only to say, hi, how are you, or something like that. However, if you have questions related to the scientific content or anything related to the presentation, please use the Q&A box rather than the chat box. This is for 2 reasons. One, because it allows the presenters and the teaching assistants to manage the questions most effectively and triage them and answer them in real time. And also because this morning I actually learned how we can save the Q&A and make them available after the presentation. So we will be doing that, and we will be posting it in the docs link of the May Institute program. So let's make sure that all the questions are actually directed in the Q&A so that we can follow them after that. And also as the logistics, so I heard this morning that at least some of the presenters today will be available at the end after the presentation. So if you would like to discuss with them something which was just wouldn't fit during the regular presentation, please don't hesitate. So this is a webinar, but we can also promote you to panelists so that you can also actually have a in-person discussion. Okay. So now, without further ado, let me welcome our speakers today. I am really, really excited to have a collaboration with Alexei's group and the FragPipe team and May Institute, because I think that this is a really important effort for our community. So let me first welcome Alexei Nesvizhsky. So Alexei is currently a professor in computational medicine and bioinformatics and pathology in University of Michigan. Alexei holds a PhD in physics from University of Washington in Seattle, after which he did a postdoc in the Rudy Abersold lab at the Institute of Systems Biology. And this was actually personally a very important time for me because Alexei and I happened to be office mates for some time during our postdoc. And I personally learned a lot from Alexei at that time. So I'm really excited to have Alexei as a guest back now. Alexei made numerous important contributions to mass spectrometry and proteomics, starting with tools such as peptide-prophet in transproteomic pipeline back then, and of course, many other tools, including his lab's work now on FragPipe. For these contributions, Alexei received many awards, including in 2017, he was a recipient of Gil Omen Computational Proteomics Award of the US Human Proteome Organization. So we are very, very fortunate to have Alexei with us today. And so I would also like to introduce 2 instructors of the course. So the first is Felipe de Vega Le Prevost. So Felipe holds a PhD in bioinformatics and in bioinformatics and computational biology in Fiocruz University in Brazil. And currently he is a research faculty in the University of Michigan. And also I would like to introduce Fengchao Yu, who is also research faculty in the University of Michigan, and he holds a PhD in bioengineering from Hong Kong University of Science and Technology. And so welcome, Alexei, welcome Fengchao, and welcome Felipe, and we look forward to hearing from you. Thank you so very much.
Well, thank you, Olga, so much for this introduction, and I'm going to share my slides. Hopefully you see them now. And slideshow. All right. So it's really a pleasure to be back. I think I've done this last year, and hopefully we'll have an opportunity to do it in person again when all this is over. But I will talk about our FragPipe computational pipeline, and I'm really excited about this. I joined Twitter so I can tweet about it. As you know, I do it with some regular frequency because I'm really excited about what we are doing and how this pipeline is working now and what we have for it in the future as well. I'll talk about quantification and FragPipe, but also I'll give you a brief overview of the whole sort of functionality of FragPipe, including post-translational modification analysis, because this is where sort of we started with developing this pipeline. About my lab a little bit. So we are a computational lab, but I'm also directing a proteomics facility here at the University of Michigan. So we try to couple computation as well as data generation, and we even moving towards sort of more wet lab proteomics as well. I want to use this very old slide. This is a pre-pandemic slides. It's actually a view from my window. Well, it's a table outside of my window. We don't have snow now, and we're not wearing masks again, so that's nice. But anyways, I should update this slide. I really want to acknowledge my lab. Again, this outdated slide. The current sort of FragPipe team, core of the FragPipe team are listed here, especially Fengchao, Felipe, Guozi, but also Danny, Dan Palaski, Kevin Young, Kai Li just recently joined the group, Jeannie and Leo more on the proteogenomics side. But I also want to really acknowledge my former lab members who contributed significantly to FragPipe at various stages. FragPipe is extremely easy to use. Hopefully some of you already have experience with that. Those who don't, I hope I'll convince you to try it after my presentation. But we have a graphical user interface, GUI, which is a screenshot of that, kind of an older version of that we see on the screen, where we have this concept of workflows, where it's extremely easy to use for you as a user because you could just select one particular workflow that you like. That's already predefined for you. For example, a DIA-based spectral library DIA analysis, or SciLac with TMT, 10, and load, and all settings are going to be already loaded in FragPipe for you. All you just need is select files, load the files, and click run. That's why it's so popular. Of course, you can customize workflows, you can save a workflow as your custom workflow, even share a mail to someone. People often contact me and I customize the workflows and email back and do things like this. So that's why it's very powerful. But in addition to that, we also have command line versions of this workflow. And in fact, in FragPipe 18, which we'll be releasing before the SMS, probably next week, we'll have a first release of what we call headless FragPipe, where you can do everything that you can do in the graphical user interface, but through the command line. That's very nice. And we have other ways of running this pipeline as well, including what we call philosopher pipeline. So you can run it on the cloud, you can run it on a desktop, on Linux, HPC. That's why it's a very powerful system, because it's platform independent, Windows and Linux, and it's scalable to very large amounts of data. So I'll talk about some advantages of FragPipe, of course, with a focus on quantification workflows that we have here. But I'll go through this post-translational modification analysis first a little bit as well, because this is where a lot of strength of our pipeline, a lot of strengths are in this sort of comprehensive PTM profiling as well. So let's talk about that part first before we move to the quantification. And again, I already mentioned that why we started developing this pipeline in a way in the first place is that this goes back to my first project in the mass spec when I just joined the Rudy Abersold lab, even before Olga became a sort of office mate. My very first project before I was officially working there was to try to understand what are all those high quality spectra that we identify, we collect, that we have no identification. Thinking that they would be a spectra corresponding to a post-translationally modified peptides or peptides harboring a mutation, or maybe novel peptides. And of course, a lot of spectra were also chemical artifacts. And it's really, now 20 years later, or how many years? 20 years later now, we have all these sort of tools, but in fact, it's because we really wanted to go deeper in the data and identify all these peptides. So this dark matter of mass spectrometry-based proteomics is why we started developing as a sort of our last iteration of our attempts to try to solve this, to develop a new search engine that could allow us to identify those peptides in a much more efficient way, comprehensive way, and also much faster way. So that's how we end up with MS-Fragger search engine that uses ultra-fast searching, implements ultra-fast searching based on our fragment ion indexing strategy that we sort of discovered and developed over the last 5-7 years. And indexing is a technology that allows us to index the data, whether it's this protein database, predicted YNB ions for all peptides, or we can index experimental data as well. So it's a technology that allows us to index the data and then search that data really fast. That's why MS-Fragger is 100 times faster than a conventional search engine. So based on that, we started, based on this advanced and on MS-Fragger search engine, we realized that we have to put other pieces in place to really create a more complete pipeline that people can use. And we developed Philosopher toolkit, and Felipe developed most of it, where we basically implemented our transproteomic pipeline components, the key components, peptide profit, protein profit, and more recently, iProfit, PTM profit, as well as various scoring modules, FDR scoring modules with all various options, razor approach, peaked FDR, sequential filters, and so on and so forth, as well as reporting and generation tools. So that's a Philosopher is a toolbox that's a key part of FragPipe as well, that sort of runs the transproteomic pipeline components, as well as FDR filtering and reporting tools. So with respect to PTMs, before we jump to the quantification, because it is one of the big parts of our success is this open massive set search strategy that we developed. And that's an alternative strategy for identifying post-translational modifications that goes beyond what has been tried before, or still tried in other tools, which is iterative or multipass searches, sequence tagging, or a dependent peptide approach that's probably best known through its implementation in MaxQuant. So open massive set database search strategy is a highly efficient strategy that's also very simple conceptually in a way, and that's why we really like that. So it's illustrated on the slide that you see here. So a conventional closed search that we're all familiar with is where you specify a very narrow tolerance of maybe 20 parts per million, for example, for a modern instrument, and perhaps allow isotope errors and most common modifications such as methane oxidation and perhaps internal acetylation. But other than that, you're only looking for those peptides. You're not looking for any other modifications or unexpected mass shifts. And that's a conventional closed search, right? So you're really looking at like, this 20 ppm around the expected mass of the peptide. Now, the open search is the sort of the opposite extreme of that, where you can have no restrictions at all. So you can search for a peptide in the database with any mass shift with respect to the measured experimental mass for that MSMS spectrum, right? So you allow any possible mass shift. So it's a complete open search. In practice, we typically do from minus 150 Dalton to 500 Dalton, or you can go to 1,000 Dalton, but in principle, it could be a completely unconstrained. And then we have this really nice strategy that we call mass offset search, where it's a combination, it's something in between the two. The closed search and open search is where we search with a narrow tolerance of 20 ppm, but around a multiple notches. For example, we can allow unmodified peptides with a mass shift of 0, but also we can specify we allow acetylation, say phosphorylation, and we'll search with a plus minus 20 ppm around mass shifts that correspond to those modifications, right? And this could be a really long list. So our common mass offset workflow in FracPipe, for example, has about a few hundred, maybe 300 most common post-translational modifications. And it's a really good alternative to a full open search. And our glyco workflow and lab modification workflow, that's now, it's a really huge field for my lab. Those areas are based on this concept of a mass offset search. So using full open search, we can search for any mass shift, and if you run it on any data, standard data, you can use it as a quality control. I do it on every data set that I'm running. I'm running a ton of data sets. I'm like a beta tester, and I should be doing a lot of other stuff, but my favorite activity is to run FracPipe, frankly. So anyways, I run a lot of FracPipe, and I do a lot of QC analysis, and you just, even if it's a regular search, triptych digest, do it, because you'll find a lot of artifacts, a lot of unexpected modifications that you can optimize your protocols and change your search parameters based on that. So this is how a histogram of mass shifts would look like for a typical open search on a regular digest. You'll see a ton of those mass shifts corresponding to common modifications, and a negative mass shifts corresponding to neutral losses or semi-triptych peptides. If you don't allow them, you'll find them through open search as well. You typically would get, a very good booster number of PSMs, 30% or more identified. So as I said, you can do it even if nothing else. You can just do it for sample QC. But we have a huge user base now, hundreds, thousands of people are using that for other things, chemical proteomics. Probably all major pharma companies now license our tools, because they do covalent modifications, they do all sorts of PTM and electrophile screening libraries and stuff. That's really great for that. But RNA cross-linking, all of that can be captured through this common mass offset search strategy or open source strategy. We extended a basic open source strategy more recently to introduce what we called localization aware open search and shifted ions. I do not have time to go into discussing this extension of algorithms of Inchao. Let this work with the help of Goetze and others. So it's published in a year and a half, 2 years ago now. But we really extend open search to also capture shifted ions. So in conventional open search, you're basically identifying a peptide from unshifted YNB ions that don't have the modification. And it's actually sufficient to identify most of the peptides, even though you lose half of the information, up to half of the information. But in the localization aware open search, it's now part of MS Fragger is a localization aware open search mode, and fully supported on FracPipe by default, actually. We also have this modified fragment ion indexing strategy using what we call shifted ion indexing. We can capture peptides with fragments that contain modification as part of the open source strategy. That really gives us a nice boost in the number of modified peptides we can identify through open search. But also allows us to localize the mass shift on the peptide, which was one of the limitations of our original open source strategy. So if you apply something like open searching to interactome data, for example, where we have bait proteins that are enriched using affinity purification, so they're very abundant. So these are not typical samples. These are samples where one protein is highly enriched in a sample. So if you look at how many PSMs we have for bait proteins in this affinity purification mass spec experiments, and here we are reanalyzing the data from the Bioplex study from Bioplex 1 from Steve Vega's lab and Wade Harper's lab. So for Bioplex 1, we reprocessed 64 million spectra. It took us only a few days. With MS Fragor, it would take you years probably with Sequest. So what's interesting is we look at how many PSMs we have per bait protein that we identified in OpenSearch compared to conventional closed search, like they did in the paper, or like most people would do still. I mean, that's amazing. We get, for some proteins, we get, 100 times as many PSMs. So here's an example of one protein, ETH-E1, where we go from 48 peptide ions found in the conventional closed search to over 2,000, almost 2,500 found in OpenSearch, right? A huge boost. So this is, there are 1,000 dots here. There are 1,000 APMS runs, 1,000 proteins used as bait proteins. So for each one, we show the number of PSMs in OpenSearch versus closed search. And you see on average, we get 400% increase. So 4 times as many PSMs. But for some of them, like this one that I show, you have 50 times as many. And interestingly, a lot of proteins that show the highest number of the highest boost, the number of peptides that we identify in open versus closed search in this data. If you do a gene enrichment analysis, you'll see that those proteins are enriched for a category, genetology category called small molecule metabolic processes, which suggests that this is the proteins that modify other proteins, including themselves. So perhaps this is why they're so heavily positionally modified in this data set. And that's why we get the highest gain in number of PSMs we have when we go to open search, capturing all those modifications compared to the closed search, looking only for no modifications or only oxidized methionine. So in the last few years, we worked really hard, and again, we have a really great team developing MS Fragor and all other tools in the pipeline. So in addition to the localization aware open search that we developed, we have mass calibration, we have search parameter optimization, we have de-isotoping, we have support for FAMES, as well as timsTOF ion mobility data. We have monoisotope correction. We have improved scoring now, improved signal processing, neutral loss removal. I mean, I have a ton of work that went into MS Fragor, as well as in all other tools. So this is really in active development. So a key tool for PTM analysis in FragPipe that we have is PTM Shuffler that Danny Geisler, graduate student in the lab, developed, or published an MCP, what is it, less than a year ago, that if you do PTM analysis, it will summarize the site localization properties for each mass shift, spectral similarity, retention time shifts for each mass shift. It will summarize the abundance of peptides in modified form compared to the unmodified form. So I welcome you to go and check out this paper. You can find and characterize unexpected novel PTMs with MS Fragor coupled with PTM Shuffler. So it's very useful. We have an example here of finding some interesting mass shifts in, for example, in a synthetic data set from Bernard Kuster lab, where with the help of PTM Shuffler, we were able to distinguish one particular modification from a neutral loss of 18 that's typical in this data to actually we can locate it to C-terminal lysine, which you think is a cyclization effect, which is a very interesting sort of unexpected byproduct of peptide synthesis that I think people should be aware of. And tools like FragPipe with MS Fragor and PTM Shuffler allows you to identify this type of events and sort them out and figure out which are neutral losses, in which are modifications that happen prior to mass spec. You can run it again for QC. This is where we run this pipeline as part of our CPTech efforts. We are a member of CPTech consortium where we work with some ridiculous amount of data, TMT data. But here we run it as a QC run using CPTech 3 harmonization data generated at 3 different sites, Johns Hopkins, Broad Institute, and PNNL, where they use the same samples analyzed through harmonized pipeline, but at different sites. And we clustered here different mass shift PTMs that we find in each site in different plexes, or if you have replicates. So each row here is a different replicate, and you see how the data clusters based on the site where the data was generated. So there are some PTMs or artifacts that are specific to each protocol, even though the protocols are harmonized. But even within the same site, sometimes we see a different sort of PTM profiles. And that's because there is a lot of variability in sample prep. For example, you could have a lot of formulation or other PTMs, maybe due to differences in Tmt labeling that happens, on different days, and so on and so forth. So we can really, with our tools, get this picture of all PTMs. And in this case, most of the things we see are probably chemical artifacts, but when you run it in biological, it's sort of, we also recover interesting biological PTMs through similar analysis. So another thing that I will not talk about at all now, except just to give you this advertisement for a huge effort in the lab, is glycoproteomics and labile modifications in general. And we extended MS Fragger with our common our master set workflow, as well as PTM Shepherd. We extended this to glycoproteomics. It's a super powerful workflow, and based on our benchmarking, it's the best tool for glycoproteomics. And others agree with that. And as part of the HUPO initiative, there is a benchmarking data set where we've done this benchmark, and you'll see the results announced very soon that we've done really, really well with MS Fragger Glyco, as well as a more recent work on estimating full discovery rates for different glycan structures that we published in MCP more recently. So this is the earlier work by Dan, and we have a new one in MCP that further extended this. So we have a glyco workflow in FragPipe that's also fully compatible with label free quantification or Tmt quantification. We have glyco Tmt workflow. So if you are into glycoproteomics, I strongly encourage you to explore this, what we have to offer. So now let's move to quantitative workflows in FragPipe, and we'll talk about label-free quantification first, and then TMT. I don't think I'll talk about SciLac, and I'll conclude with some comments on our DIE efforts and what we have now, which is actually really great. So label-free quantification. So we developed a key tool called IonQuant. That's the part of FragPipe. And IonQuant basically is a label-free quantification tool, or rather, it's the MS1 quantification tool. It fully supports SciLac and other labeling workflows as well. But talking about label-free quantification with IonQuant, basically it has three key parts to that. First, we have advanced single processing, so it's fully compatible with TeamStuff passive data, and in fact, probably most of TeamStuff users who are using label-free quantification are using FragPipe with MS Fragger and IonQuant, because it's super fast. It's completely, it's day and night compared to other tools. But also even for thermo data, we have key advantages is that we have highly efficient match between runs implemented in there. So not only optimize single processing, but we also have match between runs with full discovery rate control. And full discovery rate control for match between runs is the third key part of IonQuant. So I'll briefly go through that. So I already mentioned that it's super fast because we index the data like MS Fragger, we index the data. So that's why tracing the peaks and everything is super fast. Including for TeamStuff data, where speed is always an issue, but also for FAMES data on thermal instruments. We have FDR control match between runs, and again, we have complete support for labeling-based analysis in IonQuant as well, including SciLac. So on signal processing, I already mentioned that we have optimized peak tracing. I mean, we put a lot of efforts in trying to optimize all of that, including boundary detections, smoothing, which is always a challenge, especially for low abundance signals, And the settings in Quant are very, very flexible. So if you're an advanced user, you can really, play with a lot of these settings for how you do signal processing. But again, if you're not so advanced user, you just want to run what we think is optimal. You just load the FragPipe workflow, and it has all settings already that's set for you that we think is the best analysis. So the match between runs, the concept is well established. In match between runs, you basically have a donor run, an acceptor run. The donor run is a run where we identify a peptide from a MS spectrum that we did not identify that peptide in our acceptor run. And you would like to transfer that ID. You would like to find that MS1 peak through retention time, ion mobility, if it's Tmt data, alignment from the donor run to the acceptor run. So, and you can have maybe hundreds of runs in a data set. So for each run, you would have maybe potentially hundreds of runs that are donor runs for which you can transfer the ID. And you can either transfer from runs that are more similar to the donor run to the acceptor run, or you can transfer from any run. So all of these are highly customizable settings in FragPipe. By default, we transfer from all runs, but on very large data sets, you can restrict that to maybe top 10 runs or even less. So, this is kind of very well established. The advantage of our algorithms for match between runs here is that we have a very fast and highly efficient alignment between different runs. We use a neighbor-based alignment, and I want to give a shout out to a tool called Metamorphosis. Metamorphosis has this flash LFQ that has very nice. This had a good idea for doing this neighbor-based alignment, and we implemented that, and it worked really great. So
Alexei, there's a question which maybe
yes.
Someone is asking, what do you mean by decoy on this slide?
Oh yeah, so decoys. So these are not decoy identifications like decoy sequences in a database. Here to implement sort of false discovery, false transfer estimation for match between runs, we need to create kind of signals that we try to transfer from the donor run to the acceptor run that actually don't exist. So what we do here, and let me see if it's on the next slide. I don't really have that. But what we do is that we take a real, so in a donor run, so here we have a peak that we identified here from MS spectrum. We have master charge ratio, we have retention time of that peak, right? But we need to create like a decoy, a fake signal that we want to try to transfer and see if we by mistake also find that. So we shift master charge ratio of the peptide by certain mass. So so this idea comes from the early work from Dick Smith's lab from probably 20 years ago. Vlad Pityuk worked on that and others. So we referenced this idea in Ion Quant match between run paper. So we basically shift identified peptides by certain master charge ratio to create signals that shouldn't exist. So we shift them by like, I think 11 Dalton or 13 Dalton and try some other shifts. So those mass shifts don't correspond to any common PTM. So we basically take an MOZ that shouldn't be in the data. We take identified MOZ, shift say by 11 MOZ or 13, I forgot, and go to the acceptor run and try to find it there. And we shouldn't find it. If we find it, we call it a decoy transfer. So we basically create these decoys, try to transfer them, and then like this peptide profit, so all these things that we develop for target decoy analysis, we basically have scores of for each transferred ion, we calculate the score based on like how close they are in retention time to the expected value for the ion mobility and like the isotope profile, isotope pattern in the donor run. Does it match the one in the acceptor run? We calculate the score, and then we can see like what are the scores we get for decoy values that we transferred, right? So we have like, so the blue is the scores of those decoys, and the red are scores of target ions that we transferred, and we kind of like the model this decoy shape of the decoy distribution, and then we feed it to the target distribution to model this kind of like the negative tail here. So through this analysis, we estimate basically posterior probability of correct transfer. where basically decoys here allows us to estimate the posterior probability, and then the false discovery rate for that of match between run transfer, right? So that's what we do here. So other tools like MaxQuant don't have the false discovery rate estimation, and we do have it, and we control it at 1% iron level false discovery rate for the transfer by default, although you can change that. So that's the sort of the decoy transfer idea here. So we tested this, and I cannot see if there are questions. I cannot see them because I only see my slides. So let me know. Thanks for bringing this up. Okay. So we tested this on a whole bunch of data sets, of course, and like everyone else, we have to compare with other tools. And MaxQuant, frankly, is an easy comparison for us. So anyways, this is comparison with MaxQuant, match between runs, which does not have FDR control. But this is a data set. This is a sort of a popular way of doing things. We have 3 organisms mixed at different proportions in a sample A versus B. So we have A versus B with a human 1-to-1 in both samples, and then we have yeast, and that's 2-to-1, so log-fold change 1 in sample A compared to sample B, and we have E. coli, which is 4-to-1 ratio, mixed at 4-to-1 ratio in sample B compared to A. So it's a negative log2 change of -2. And we quantified more proteins in IronQuant with match between runs and MaxQuant. You see the numbers here. But importantly, as you look at the number of outliers, visually you see that we have much fewer outliers, like the tighter distribution of one, of around 1 for human, and less outliers for both yeast and E. coli, compared to, this is the team stuff data, compared to MaxQuant here, and it's visualized using LFQ bench. So we have excellent quantification performance compared to MaxQuant. On single cell data, for example, this is why most single cell groups now adopted our pipeline, including Inju Lab from PNNL. So this is his data. We convinced him that our pipeline is great by showing him his result. His data analyzed through our pipeline. So they're our biggest fans now. So this is Orbitrap Fusion Lumos for replicates with 50 cells with this boosting channel. So you could use, if you do a label frequency, LFQ single cell data, you can use 50. You can do 50 cells, and then transfer. You call them library runs or boosting runs, and through match between runs, you transfer IDs from 50 cells to single cell data. So 50 cells is library runs for replicates, and then you have 11 replicates, so single cell data, but you also have tests for 3 cell data and 10 cell data. And again, we control here with 1% FDR. We don't know what FDR is for match between runs and MaxQuant. It's not controlled. So let's look at the single cell data. Now, obviously, with match between runs, we quantify a lot more proteins and peptides in single cell data. So so here we have 11 dots. No match between run. We have only 250 proteins on average, goes to 750 with match between run, and of course a similar boost for MaxQuant as well. But we have more than in MaxQuant. But you see, 0 cell is a good control here. Match between run in MaxQuant kind of like boosted too much the number of IDs in 0 cell data, where because we control it in an IronQuant, we don't have that boost in single cell in 0 cell data. 0 cell is blank data. Ideally, we shouldn't find anything. It's not a perfect control, but still, I'm glad to see that we are not boosting our IDs with match between runs in 0 cell data as much as MaxQuant. Again, it shows that we are controlling the FDR, and we still have overall high number of proteins quantified. So that's the single cell data. And we demonstrated this across a wide range of data sets. Others use this now on TeamStuff data on thousands of runs. There is a Cell paper from a consortium, COVID consortium in England, where they run our pipeline I mean, the pipeline is adopted now, by many labs, and there are a lot of independent benchmarks that people published, sort of that you can go and look up. Because everything that we show, of course, we're a little bit biased. So we should look at what others get with our pipelines, and you'll be pleased to see that that actually works well. And the speed has always been an advantage for us due to indexing. So again, MaxQuant is probably not the best comparison because it's just so slow. Perhaps they improved it recently, but even against other pipelines, we do really well. So this is the comparison against MaxQuant here, where we have you go from days to hours really when you run this. So more recently, we added a very important module in FragPipe. Before I continue with quantification, I just want to mention that we also added rescoring based on deep learning called MS Booster, led by Kevin Young in the lab. Work really great. It boosts the number of IDs, and it helps us to boost the number of IDs, especially in cases where the search space is large, where the gray zone is bigger, such as HLA, peptidome data, or non-specific searches of all kind. But it works for any searches, really, even basic TMT searches. Label-free searches. So again, a preprint is coming soon, but it's fully functional in FragPipe, and in fact, it's already default for most workflows, called MS Booster. So this is just a slide illustrating that we gain IDs in single-cell data sets, but we gain across the board in all data sets, and especially in HLA peptidome data. That's where we have a huge boost, as well as in DIA workflows that we also gain a lot. So let's
Alexei, before you move on, another question related to the comparison with MaxQuant. Dmitry is asking, can you say that the difference was seen in low abundant proteins? And if yes, have you, or is it possible to manually verify what's going on?
Well, the difference with MaxQuant, of course, the low abundance proteins, the proteins that are quantified by a lower number of ions, is where you gain the most. Because if you have more, tools like IonQuant, IonQuant can be run with MSTATs, for example, too, and MSTATs will model peptides, right? We have something in IonQuant that combines multiple. We have this maxillary normalization actually in IonQuant. So the more peptides you have, the more robust quantification will be. MSTATs, of course, with multiple peptides, will be able to eliminate outliers. So the challenge is always for single peptide identifications and quantification. And we think that through improved signal processing, we have better quantification. But importantly, we control the full. Again, I think the key advantage is that we are controlling match between run transfer FDR, right? So we are controlling transfer of those false IDs, and they are the problem. So if you're transferring a ton of quantification correctly, from a wrong peptide, and you're not able to eliminate this at a later stage, because all you get is just 1 or 2 peptides, you're going to get a lot of false quantification values, right? So that's why we're doing better. Now, manual quantification, well, sure, you can, if you're an expert person, you can look at the data and you can convince yourself that the tool is doing well. You can only do so much manually. So we use strategies such as using LFQ bench, using these organisms with multiple organisms mixed at known fractions, right? Or replicate runs, CV cross runs. You can utilize those strategies. But if you're an expert mass spectrometrist, you can of course convince yourself by looking. You can load in Skyline, in other ways, and see if we are quantifying it well or not. And in fact, we are working with many groups. We have a paper published in Molecular and Cellular Proteomics just a month ago in collaboration with Kerry Buckwith's lab at UCLA, where they extensively used compared IonQuant against Skyline and benchmarked the tools against each other. They looked at they used Skyline a lot, and they benchmarked IonQuant, and both tools work well. And IronQuant was extensively benchmarked against Skyline. So I welcome you to look at this. Kerry Bacchus, Google me and find that it's a chemical proteomics application, MCP from a few months ago, a benchmarking against Skyline. So exactly like you were saying, a lot of manual processing, as well as a lot of manual analysis, a lot of benchmarks with, again, 4-to-1 labeling and 2-to-1 labeling, 1-to-1 labeling, and so on and so forth. Any other questions?
Yeah, just a logistics question. Several people asked if it's possible to have the slides after the presentation.
Yeah. So Olga, just let me know. I think I shared just PDFs, right?
Just send this to me and I will share. Thank you.
So I assume that the video is also going to be available too, right? Of that.
The video is available, yes.
Right. All right. So if no other questions to the TMT I-track pipeline. So this is, we worked a lot on this because we are part of the CPTAC consortium, and we are really processing a ton of TMT data. So we worked on this for a while. And our pipeline has been used as part of 4 or 5 now landmark papers published through CPTAC consortium. Some of these papers listed here, but of course also non-CPTAC data sets as well. And again, this is where MS Frager, Philosopher, now we use percolator more rather than peptide profit, but PTM profit for localization, protein profit, protein inference, quantification, extraction of quant, and then we have TMT integrator that's as part of FragPipe, but also MSStat is absolutely a great tool for taking PSM TSV files from the pipeline and running through MSStat. So we have options. It's really nice to have great options for analyzing the data. So we have just to go through various steps, because every tool would go through that. We have TMT integrator that kind of pre-calculates some of these filters. So TMT integrator takes a PSM.tsv table from every data set, like MSStats would take PSM.tsv file from every plex, right? So each data set would have multiple plexes. If you have only one plex, that's of course, you just have one PSM table. If you have multiple TMT plexes, with or without bridging channel, then you'll have multiple PSM tables generated by FragPipe. And then you'll have to go through certain steps to filter the PSMs and then assemble them, group PSMs into proteins or peptides, and aggregate the information and generate the reports, right? So, and the common steps are, reference normalization if we have a bridging channel, grouping, outlier removal, aggregation of PSMs, and then normalization. So, typically the PSM filters that we apply, and I think MSStat's applying some of these filters, in the same way, but it also has more advanced modeling for some other things. So our basic PSM filters, our PSM tables we compute. Of course, we apply FDR-based filtering in the first place before we generate PSMs. PSM.tsv file is all quantification, but we calculate isotope purity score. We apply intensity cutoffs to eliminate PSMs with like less than 5, intensity that's less than the lowest 5% of the intensity in the data set. Sort of we can eliminate by default. We use 2.5% for phosphoproteomics by default. The PSM tables have everything, but the filters that we apply in TMT integrator, that's where those filters are applied. We also select the best PSM among redundant PSMs per fraction in TMT integrator in whole proteome data. And then we group PSMs based on a specific index. And our index, we have multiple protein, peptide, G, site level index if it's PTM data. And then, we do outlier removal. If you have multiple PSMs for the same protein, we kind of see that the ratios, after we calculate the ratio to the reference channel, we aggregate PSMs to the protein, and you kind of try to establish sort of a median level across all PSMs, and in doing so, we eliminate outliers before we calculate the median. So, once we eliminate the outliers, we take the median of PSM level ratios to calculate the corresponding sort of ratio of the protein level. So it's a pretty simple strategy, and MS Stats would do a more elaborate modeling here, but Tmt Integrator by default does what I described here, basically aggregation of PSMs to a particular level, some normalization, outlier removal, followed by additional normalization after the fact, of which we have medium centering and more advanced, more aggressive normalization that we no longer recommend to do. Our median centering is sort of a standard normalization that we do. And you also have an interesting step that I don't have time to explain, but we have conversion of a reference. First we have a reference to a ratio to a reference sample, a bridging sample that we have. If you have a single plex, we use a virtual reference approach where kind of it's a reference to an average of all samples. But if you have multiple plexes and you need to merge them, the best way is to insert a reference sample that's a pool of all samples. Now, if you don't have that, you still can use a virtual reference approach. But anyways, everything is converted first for each PSM as a reference, as a ratio to that reference, whether it's a pooled sample or a virtual reference. So our final, our tables aggregated to the peptide or protein or gene level are first also ratios, right? It would be like for each protein, there would be a ratio to a reference. But to convert it back to the absolute abundance intensity, we have this one additional conversion step where we estimate abundance of peptides and proteins in the reference sample through several strategies. We use MS1 intensity for our default strategy. We calculate MS1 intensity of each peptide ion, apportion that intensity according to the PSM level ratios, reporter ion ratios, and calculate kind of like the MS1 intensity of the peptide and then the protein in a reference sample in each plex. And then we aggregate measurements, reference sample measurements across all plexes to get what we call reference intensity. So at the end, we calculate what we call abundance tables, where ratios to reference for each protein in each sample gets converted to abundance intensity, basically, in that sample. So we have a ratio table and abundance table. An abundance table is kind of like the absolute abundance in a way in each sample. So if having absolute abundance is useful for downstream statistical methods that would like to know what's the abundance of the protein is, not just a ratio, or if you want to correlate, say, against RNA-seq data for each sample within each sample, then you really, ratio is not going to do it. You have to have absolute abundance estimated, and for some other applications as well. So anyways, again, we, as usual, I'm running a little bit behind, so I'm going to probably cut the IA data part a little bit, but of course we compared it against a whole bunch of other tools, and you can find benchmarks published in literature as part of our papers, as well as we compare it in a collaboration with Binzang, lab at Baylor, for example, for phosphodata. We have a paper on MCP a few months ago. We use some deep learning based benchmarks to evaluate Tmt Integrator in FragPipe, or rather the whole pipeline, sorry, not Tmt Integrator, the whole sort of our pipeline against a whole bunch of other tools, including MaxQuant. Here, this is our comparison for the CCRCC dataset from CPTAC, where you run through Tmt Integrator and MaxQuant would take you like 2 weeks to run it. Tmt Integrator will take you like 7 hours on a cohort like this. It's a huge cohort, 23 plexes, 24 fractions each, and still you can analyze it in half a day with our pipeline and 2 weeks with MaxQuant. But anyways, we have a much better correlation against RNA-seq data is measured, as an indirect measure of accuracy of quantification. Not to mention that, again, we quantify more proteins. We also, as part of CPTAC, we run this on 15 cohorts, so the pipeline scales to a ridiculous amount of data. So now it's like 15 CPTAC cohorts. It's just gazillion of data. Another advantage is that, again, we generate reports at various levels. Again, I don't have time to explain this now. Perhaps Felipe and Finchao can explain that later when they show the output. But we generate reports at gene, protein, peptide level, multi-site, single-site level. Single-site tables are if you do like a phosphoanalysis, and you want to only keep sites with confidently localized sites like PTM profit probability of 0.75 or above. So those will be only present in the single site data. Multi-site data will contain unlocalized site as well, and so on and so forth. So and here is some information about how we generate those indices. In the case of phospho data, again, I don't think we have phospho really. I'm not going to explore as part of the sort of hands-on later that Felipe will do with Finchao. So. But all of this is described on our website. So we generate really, like I said, PTM level reports, where in a multi-site report, we keep all configurations localized, unlocalized, partially localized. But in a single site report, we'll only keep localized sites and only at the level, only like a one row, one site. Where in a multi-site report, for example, a peptide that has 2 phosphosites, so the localized will be a different entry from the same peptide with only one of the 2 sites localized, or a monophosphorylated peptides. They will be all kept as separate entries in the site in a multi-site report, and they will be collapsed to like a single entry in a single site report, one for each site, and only if it's localized. And this is a duplicate of the slide. You just saw that. We've done a lot of benchmarks in a non-CPTAC data, for example, looking at again in a human plus E. coli data set. It's an E. coli data set where there were human protein spiked in at known ratios, right? So again, we compared against Protein Discover, MaxQuant with different settings, and to demonstrate, and we have MS2 data here and MS3 data. So this is something that will be, it's part of the manuscript that we are writing now, and that you should see soon, hopefully it's a preprint as well, hopefully in the next month. But anyways, we show that both in MS3 data and MS2 data, we have really a good performance. Of course, we still have the ratio compression effects in MS3, like all tools have, but we also have various ways to minimize that. And we have comparison against Protein Discover or MaxQuant to show that we quantify more proteins and the accuracy is comparable or better than other tools. So it's pretty much 1 minute until the end of the hour. So I will just very briefly tell you that in addition to TMT, we fully comport DIA quantification. It's just, this is where we put a lot of efforts in the last few years. If you're familiar with our work in this area, that we sort of pioneered this direct DIA approach with DIA Empire, as well as the hybrid spectral library building approach, when the libraries are built from DIA data directly combined with DDA data, if you have that, to build a single library. So this is what we sort of published, a while back. People first did not believe in direct DIA, but now everyone thinks that's a really good strategy. So FragPipe supports fully DIA Empire-based workflow for building hybrid libraries. And now it's a one-stop workflow. We added Diane from Quantification in collaboration with Vadim Demichev. Diane is really a great tool. It's a part of FragPipe. So it's just with a few clicks you can load DIA data, you can load DDA data if you have. And then if you use DIA Empire workflow, it will run DIA Empire, extract through MS-Frag spectra, will go through MS-Frag research, MS Booster. DDA data will go through the same. Protein Profit will all combine it into a single library with Easy Pqp, FDI filtered, and so on. And then we'll go to DIA data with Diane and extract quantification. So one-stop analysis. But before you get all too excited about DIA Empire, we have a new tool called MS-Fragger DIA that now MS-Fragger can do direct DIA analysis directly, and the results are just outstanding. So again, all benchmarks, not only we of course beat our own tool, DAO Empire strategy, but we also perform really well against all other tools, including Spectronaut. And again, you can do it with just a few clicks, load DDA, load DIA. MS-Fragger will recognize what's DIA, well, you have to annotate them to DIA or DDA, but MS-Fragger will automatically run DIA data through MS-Fragger DIA tool. The DDA will go through the conventional DDA algorithm, deep learning, rescoring with deep learning with MS Booster, percolator, rescoring, peptide profit, easy Pqp for spectral library building. And then given that combined hybrid spectral library, again, Diane will take the DIA data and will match against that library and will extract quant. So this is how it will look. You'll see that in FragPipe 17, it's already available. I have a DIA Empire already separate. And Diane is a separate tab here, DIA Empire as well. But all you need to do is to load like DIA spec lib quant workflow, load DIA and DDA data, annotate DIA runs as DIA, DDA is DDA, and then click run, and the whole thing will run all the way to through Diane. Now, if you only want to use, for example, some runs for DIA for quantification and not for building spectral library, you can annotate them as DIA quant. In that case, the DIA runs will not be used for spectral library building. If it's as an option. If you do GFP, this is actually great. The stuff that Mike Macas, developed, really great ways to generate DIA data with a gas phase fractionation strategy for building the libraries. So you can load gas phase fractionated data, set it to GFP DIA type, and load wide window DIA data, annotate as DIA, and then build a library from all data combined. It works great as well. So I had some benchmarks. Let me skip that. Just say that, again, on this one is a particular phosphor, very deep phosphor data set that we, compare it to Diane, library free. So Diane also, and Diane is a really great tool. All tools here are great. Spectronaut is also good. And Diane is good, all have advantages, but of course, we have to show that we do better than other tools, right? Otherwise, why would I show this? So don't trust me, go and do it yourself. Don't trust any bioinformatics group, whatever they show you, run it, compare yourself, and then decide for yourself. But in our comparisons, we do really well. So here is this number of phosphosites quantified compared to library free. If you run a library-free Diane followed by quantification versus MS-Fragger based library building followed by Diane quantification. So this is MS-Fragger DIA plus Diane. So we can quantify more peptides, certainly more than Spectronaut, and outperform DIA Empire as well. But DIA Empire has still advantages for certain applications. Don't have time to discuss this, but there is a lot of advantages for DIA analysis with DIA Empire as well. For like non-specific searches, for example, still DIA Empire based. I recommend it. Now, for long input single cell data, we are now writing a paper on MS-Fragger DIA. So, again, we show against Spectronaut, for example, on 5 cells, 10 cells, 50 cells. We also have single cell data where we show that through our MS-Fragger DIA strategy, we significantly outperform Spectronaut, not only as far as the number of IDs, but we also get better quantification. And that's because Diane is absolutely outstanding tool for quantification. So it's that. So here I'm showing 5, 10 cell data from a recently published data from our collaborators from UG Chen lab. I just spent a month in Taiwan, again, met with them, a really great group. So we are also working with them, adapting our tools to the single cell low input data. And they have a really nice technology for single cell analysis, DIA based, that we are collaborating on and running our tools on the data. It works well. If you're on Twitter, and you should go and follow me on Twitter, because I advertise on Twitter like there is no tomorrow, because I'm, what else is Twitter for, right? Other than just shameless self-promotion, right? So Here is, I occasionally download the data sets and run through the pipeline and see the results. So I got excited about this ultra deep DIA publication from a few days ago. Here is the reference in Journal of Proteome Research. So I downloaded, run through MS-Fragger DIA, three replicates of cell line, 120-minute gradient, three replicates, and 10,000 proteins quantified, over 8,500 quantified with CV less than 10%, with MS-Fragger DIA coupled with DIA. Works well. Team staff data, maybe you've seen a paper from Matthias Mann lab on single cell proteomics. So they adopted FragPipe and Diane. So basically they adopted our pipeline, which is, I don't need to go and show you our results. If Matthias Mann lab thinks that our pipeline works well, it probably works well. But again, don't trust what I'm saying. Take your data, run it, and compare with other tools, because other tools are also good and see what works best for you. So interactome analysis, the output from our pipeline fully compatible with our reprint resource. So you can load everything. If you do interactome studies, take the output from FragPipe, quantify it using spectral counts or ion quant intensities, load and reprint reprint the Ptms for interactome analysis and scoring. As well as full support for chemical proteomics workflow, which is something we're very excited, but it's way too complicated to explain, especially because I'm out of time. So, again, thank you for listening for so long for me. I talked about FragPipe from our workflows for post-translational amplification analysis to quantification that we do, whether it's label-free quantification or TMT or DIA. But we also support SciLac and advantages of the pipeline when it comes to speed and accuracy, and all can be downloaded from this link. FragPipe 18 coming out soon. And again, sincere apologies. If you know me, I'm always run out of time. This is my problem. I should do something about it. Well, maybe next time. There's always next time, right? So I'll stop here, and if you have any time for questions, I'll be happy to answer them.
Well, thank you so much, Alexei. I know you have to leave soon. Can you tell us how much time you have?
No, I have time. It's how much time you have. I'm okay.
Okay. So, well, there's a couple of questions. So, like, I don't even know where to start. So maybe talk about the PTM identification with MS-Fragger, FIA, and Diane. And for example, deamidation. If you can comment.
Deamidation. So with what quantification workflow? So, we support DIA, TMT, label-free, all of that, right? If the MS-Fragger is just a search engine that will identify them, identify the phosphopeptides. Now, then how you could generate this? These are sort of the reports, right? If it's label-free, we have site level reports that are generated by ion quant. So maybe Finchao can show you that. Ion quant will take, if you run PTM profit, ion quant will look at the localization probability of 0.75 for deciding which sites are localized, and will generate the reports. If it's a TMT workflow, if it's PTM profit that's used for localization, TMT integrator will use that, and you'll have these reports. If it's SciLac, we have SciLac as well. Ion quant does SciLac and regenerates the reports. And if it's DIA, that's a little bit of a partial solution right now. We'll build the libraries that contain the phosphosites, and Diane will, then Diane has ways to quantify the phosphopeptides and report some additional information that's useful for knowing, how confidently it was with extracting fragments specifically for those phosphosites. DIA with PTM analysis is still quite challenging, actually, whatever the pipeline you use. So it's a work in progress, I would say, but certainly Diane is doing a pretty good job already. and our part on Spectral library building part will build the libraries for phosphopeptides.
And is there a limit on the number of PTM for a search? I guess it relates to DIA.
Well, for the convention, if you do the regular DDA search, DDA data or DDA or DIA, a closed search, right? We have 7 or 9 variable modifications or something that we allow by default. More than that, but you know, PTM analysis is our bread and butter. So we work with groups to do all sort of crazy stuff. That's why we have common mass offset workflows. We have labile workflows. You know, we can search. Maybe if Inchao can, you know, you can show and I can stay a little bit maybe in that comments too. You know, if you open the workflow, if you open the frag pipe, you can, you know, you can open the a mass offset workflow. You can put common mass offsets, like you can put multiple mass offsets there. You know, even with phospho, we often do it through the mass offset strategy because phospho is a labile modification. So frag pipe is super powerful for that because there is a lot of advanced options. So it supports all the basic stuff that you know from any other search engine. You know, put 7 or 9 variable modifications and do it that way, right? But if it's more than that, I suggest you start learning mass offset workflows, how to specify mass offsets and go through that, because then there is no limit, right? Then or you can do the open search, then you know you can search for everything in one go.
And there's a question that came up when you talked about TMT. So could you comment on the number of identifications between MS2 and MS3?
Okay, well, the number of notifications always drops if you do MS3 based. I mean, you know, I want to say probably the number of PSMs maybe will drop, you know, 20-30% at least. The number of proteins that drop will be less, you know, maybe 15-20%. Yeah, there is certainly a drop in the number of IDs if you have to acquire additional MS3 data, right? That's why CPTech, for example, uses MS2 only approach. Now, the price you pay is that, of course, there's a much bigger ratio suppression effect. So with MS2-based TMT analysis, you know, if it's a 10-fold change, biological change, you'll be likely to get, you know, 2-3-fold change from TMT analysis, right? You know, MS3 would allow you to get a more realistic fold change, a more accurate fold change. But the price you pay is the loss the number of IDs. I'm not a big believer in real-time search engine. For I should rephrase it. I mean, I think we need to explore the role of real-time search as part of MS3 workflows. I don't believe in it. No, I'm sorry for saying that. I think it has limitations, but it has advantages too, right? As a way to boost the MS3 workflow. So there are pluses and minuses. So I think you need to decide what's more accurate, what's more important for you, the accuracy of quantification or the number of IDs. In our facility, for example, we mostly do MS3 based because we don't care how many proteins you quantify. We want to have more accurate and more reflective fold change, reflective of the biology. But a lot of times, you know, you just want to differentiate between changing and not changing, and MS3 workflow is just fine, and you get more IDs.
Thanks so much. So we actually have a few more minutes. So for the participants, this is your chance to ask questions to Alexei, or if you maybe would like to hear a little bit more details on some of the previous slides, that is the moment. So let's see if you have. So I think we have a few more minutes on the schedule. So let's give it a shot. And maybe while we're waiting for more questions, Feng Chao and Felipe, could you maybe say a few words about what is planned for the hands-on more specifically? So what type of workflows also, so that everybody can have a preview?
Yeah, absolutely. So we're going to show how to, well, we're going to show a few things. First, we're going to demonstrate how to find our tools and our programs on the internet, how to work with the websites, how to download them. Then we're going to show how to configure a RAM using a simple DDA Tmt MS3 experiment. How to operate the program, how to put all the parameters in place, and then we're going to go through the output files and explain how you can use them.
And I don't think we have anything on PTM. Do we have anything on PTM? Well, I guess this is on quantification, right? Focused. So so that's what you have. Mm-hmm. Well, you know, it's the good thing about FragPipe is that it's very intuitive. So everyone who would like to use it for a particular workflow, and again, our FragPipe is knife sort of proteomics workflows, because we support pretty much everything. We don't have cross-linking now. Maybe we should, you know, at some point add that. But you know, you just select the workflow that most similar to what you want to do, or exactly what you want to do, load it, and the things are already set for you there. And TMT is just one of the workflows, or rather, we have multiple workflows, because TMT 10, 16, TMT phospho, TMT ubiquitination, and so on.
So going back to MS2 versus MS3, can you comment on why MS3 spectra are better for quantification?
Well, yes, because so how we quantify in TMT is that we have this reporter ions in the mass spectrum that basically, in principle, reflect abundance of you know, reporter ions, you know, for a particular sample. That's what it should be measuring, the abundance of that peptide in that sample. But the problem is that we are not isolating just one peptide for MSMS. We are isolating a whole bunch of ions with a window, you know, of, you know, say ±1 delta, right? You know, you can narrow it down with Eclipse Explorers. You can successfully or Lumos narrow down to 0.8 delta, and with all the fusions you have to go with higher windows because the ion optics wasn't that great. So, but there's still a pretty wide window. So you're actually isolating not just the target peptide, but possibly some co-fragmenting peptides. So you're identifying the peptide on which you acquired the SMS, but the reporter ion intensities reflect the presence of those co-isolated ions, other peptides, and their abundance, right? So they also contribute to those reporter ions. So in MS, and that's why you have the ratio suppression effect and some inaccurate quantification in MS2 based TMT data. In MS3, you take one additional step of actually quantifying from MS3, where you will select Y and B ions, or expected Y and B ions, from the peptide on which you're acquiring the spectrum, really, right? And collect MS spectrum with MS3 spectrum on that peptide, and the reporter ions in MS3 then will be really reflecting just the peptide that you identified. I mean, that's also not perfect. There is also some interference, but by this additional isolation, sort of the isolation step, you're removing some of these interferences from quantification.
Sounds great. And kind of along similar lines, how reliable is DIA for identification of PTMs, in particular for weak ions in the phosphopeptides and mixed spectras, given that we have a wider isolation window?
Yeah, so, I mean, we get a really good number of IDs. How reliable, if the question is the number of, I think the accuracy of quantification, that's where it's a really good question. So I don't have a really good answer right now. I have not seen a lot of good benchmarking data sets to really look at the, you know, the accuracy of quantification of, say, for phospho data, DIA versus DTA. You know, I've seen the number of IDs in direct analysis. You know, if you look at the signaling pathways from, say, maybe differentially expressed phosphosites in DIA versus DDA, you know, those would show a comparable picture, and the number of missing values would be less by the nature of your sort of more targeted extraction of quantification. But I don't know. I think it's a work in progress. I think more needs to be done to see how accurate we can be with DIA specifically for PTM containing peptides.
So another question, Silas is asking, for curiosity's sake, how much of the data from a typical proteomics run, for example, DDA on Orbitrap, is explained by standard search settings? Or how common, abundant, and common is it to find PTMs in typical standard proteomic data?
Yeah, so a really good question. This again goes back to where I started in my career in this field of, what are those unassigned high quality spectra? And that's, again, going back 20 years ago, LTQs, not LTQs, LCQs, like 2000, 2001, right? You would probably get only 15% of MSMS scans identified with high confidence, right? 10, 15%, if not less in some challenging data sets. But a lot of that was, a lot of not identified things was just noise. These days, I would say probably we're getting closer to 30, 40% of spectra that can be identified in a regular search with only looking for, well, maybe 30% looking for common modifications like oxidation and so on. If we do open search, we'll boost the numbers, right? Because we get all these chemical artifacts, as well as capture non-specific, the neutral losses, tryptic peptides, and so on and so forth. And probably will get to maybe 45%, 50%. Still a large portion of MSMS spectra are not matched. Now, out of that half that's still not matched, there is a lot of low quality data because the instruments are fast. So, you're often just acquiring MSMS on just nothing, right? Because there was no a good strong signal sort of to acquire MSMS. But there is also a lot of high quality spectra still left. So even with open searching, with everything, we go to, okay, so maybe you explain 60% of all spectra, but probably explaining 70, 80% of high quality spectra. So what are those extra 20%? Yeah, they are mutations. So, open search will find them with some mesh shifts that we may not be very confident about them in open search, and that's why they're not passing FDR because we're pretty stringent. But we've done. It's a big progress in how much we can identify now, especially with open common method search strategies. But still, there is more to identify. With improved scoring, with improved sequence database generation to capture those variant peptides and novel peptides. I think there is still room to gain, room to improve.
So somewhat related, now when things get more difficult, in particular in single cell workflows, have you tried identifying PTM?
Yeah, of course. I mean, the difficult is our sort of, we're not really interested in, for simple stuff, you can, just go and run your favorite protein discovery tool if you have it. Actually, I should take it back. Now our pipeline is so good that, you should run it for everything, not only simple stuff. But what we're really interested in is the difficult stuff. So, when we get the data like single cell, the first thing, I usually try to do is to run open search. And it's, we find a lot of interesting stuff even in single cell data. So some of that, of course, you find post-translational modifications too. You do see phosphorylation and in ubiquitination, even in single cell data. Not a whole lot, because of course you're not starting with a lot of stuff to begin with. But we see some interesting artifacts because single cell workflows like nanopods or some, they require different type of materials, different tubing, different. So we see some polymer contamination, for example, that you would not see in a traditional workflow necessarily. So we see some interesting mesh shifts. So I gave a talk on one of these. I think I gave a talk on ASMS single cell workshop, and I think I'll speak again in Boston actually in a month on single cell. I may show some slides of things that we find in single cell data, and some of them artifacts that are just related to the type of tubing that you use in single cell workflows to minimize the sample losses, or some type of reagents that people use. And I talked to Ryan Kelly about this in Inju and Thermo people. So they all like scratch their head. Oh, okay, that's where it's coming from. Okay, so yeah, there is a lot of interesting stuff. So if you get our pipelines, you can see all of that for yourself.
Well, and one more question, going back to TMT. So there was a paper where authors showed that if you take TMT balancer I and conjugate it to peptide for quantification, it is better than MS3. Do you happen to have an opinion about that?
Yeah. So I talked to
So
talked to the authors of the.
There is a link to this paper. I can put it in the chat.
I know who you're talking about. I'm just I need to go back to. I'm going to stop sharing so I could see it in the chat.
Yeah.
Right, Martin. I talked to him at his poster a few years ago. I had a really good discussion with him. I think these guys are doing really interesting work. So we haven't looked at that much because my understanding is that the commercial versions of the instruments somehow are not supporting this type of workflow really well yet. Well, anyways, I think this is exciting. This is interesting. If people start using it or if the data is available, we'll be the first one to look at that, or not the first ones, but we'll be super interested in looking on that. So if someone has good, interesting data already, if it works, if you can generate this data, send us email. We would like to take a look at the data and see what we need to do to support it.
Okay, so there's a couple of more technical questions, but I think we can answer them just in writing. So I think this is a good time for us to take a bit of a break. So I would suggest that our presenters, if you see questions which you can answer in the Q&A in writing, please do so. And otherwise, we will take a 15-minute break and we will be back at 12:45 Eastern, so quarter before the hour, wherever your time zone is. And then we will continue with the hands-on by Felipe and Feng Chao. And Alexei, if you can stay around.
I will log out, but then I'll try to come back. And thank you all again for organizing such a terrific course. I mean, I'm glad to see so many people participating. And thank you all for your interest. And I hope that some of you will be users of new users of our pipeline.
And maybe I can volunteer if you come to ASMS, find Alexei and find his team.
I have a poster on Monday. I'll be by my poster, so come and say hi. And yeah, I have a poster on Monday, and then we have a few other posters on Glyco on Thursday, and Felipe. No, not Felipe is not going to be there. The Feng Chao has a poster on something else actually. So, anyways, we'll be there. Find us.
Wonderful. Well, thank you so much. So don't go too far. We will be back quarter before the hour.
All right.
Thanks a lot.
All right, now it's 12:48. Let's see, Feng Shao is there. I think Kevin is also there. I don't know if Olga is going to be joining us for now, but I think we can start, right?
That sounds good to me.
All right, so let's see here. So I'm going to start sharing my screen here. Okay, so I'm going to start showing how you can you can find more information about our group, about the tools that we make. I'm going to show you how to locate the programs and how to download them, how to get them ready in your computer. Then
Hey, Felipe.
Yep.
Someone raised a hand.
I cannot see the chat window because I'm sharing right now.
Dmitry. Okay, allowed to talk. Dmitry, you can talk now.
It was a mistake. He put it in the chat.
Okay.
Okay, that's fine. All right, so, and then okay, so we're going to show how to run the programs, how to get them configured, and how to run them, and then we're going to show how to understand the output files. All right, so first we have a website here. It's called nativelab.org. So here you can find all the basic information about our group, especially the tools that we make. So we have a software tab here where you can find links to pretty much all the tools that we are developing right now. But most of them can also be found here on our GitHub account. So we have this GitHub page here. All the public projects are available here. So you can grab them, all of them on the GitHub, with the exception of Fragger, and I'm going to show you how to get Fragger. I'm going to show you here more specifically about FragPipe. So if you click, let me go back here. If you click here on the FragPipe repository, you're going to be redirected to our website where you can find pretty much all the information you need about how the program runs, some technical details. We have some tutorials, some workflows, some resources to show you how to read the output files, how to understand the workflows, and how to convert the files to the open format that we need. So pretty much everything can be found here. And if you click on the download button, you're going to be redirected to the place where you have to go to get the program. So now I'm assuming that everyone already has have the program in their computers. You don't necessarily need to follow the hands-on right now. So this is going to be recorded and broadcasted later. So you can just watch the presentation now. And if you want, you can try to follow up what you're doing later. Okay. So back to our GitHub, we also have links to other projects. So here, for example, this is the Philosopher website. You can find the source code. You can also find information on how to run the program, how to use all its options. And one important thing is, let me go back here to the FragPipe, is how to report back issues, or if you have questions or any requests. So here at the FragPipe website, we have this issues button here. And to use that, I believe you need to have an account. So you need to create a GitHub account. So if you click here, you can see every discussions that we're having right now, some open issues and things like that. And if you want to report a problem or ask for help or just get in touch regarding something in particular, you can click on this green button here called New Issue. And I'm not signing right now, so it won't open for me. But you have to click there, and then you will be able to report the issue you're having, or if you have any questions, or if you want to request for some kind of feature or something like that. So I'm not going to download the programs. I believe you already have them there. Some of them can be fetched on using FragPipe. And now I'm going to stop sharing here to allow Feng Xiao to share his screen. So Feng Xiao now is going to open the program, and he's going to load an experiment, and it's going to show you how to go step by step, configuring the program and executing and executing its options, so we can have the output files at the end.
Sounds good. Thank you, Philip. So, okay. So as Alex and Philip already presented, we have this FragPipe things, and I will show you how to use it with little effort to setting the parameters. So right now I have this latest released official FragPipe 17.1 in my computer. So you just go to the folder and go to this bin folder. If you are in the Windows, you can double click this one. If you are in the Linux, you need to run this shell script. So after
Feng Xiao, I think you need to share your proper screen.
Oh yeah, we are looking at your Google browser.
Oh, really?
Or the desktop, yeah.
Oh, sorry.
Okay, so yeah, just taking this as an
Okay,
opportunity, you can run FragPipe on Windows and Linux machines. We don't fully support Mac for technical reasons, but the program should be fully functional on any Windows or Linux version.
I'm sharing the right screen, right?
Yeah.
Okay, thanks. So, okay, let me say it again. So after you download the FragPipe, you will have this folder, and then you go to the bin folder here. So if you are on the Windows, you can double click this one to open the FragPipe, or if you are in the Linux, you can use this one. So after double click it, you will see the FragPipe opened. So initially there will be those three boxes will be blank. Because license issues, you have to download the FragPipe by filling these informations and check those agreements due to the license issues. After you fill those boxes and click yes, FragPipe will download it for you automatically and put it here. And the philosophy is easier. You just need to click automatically. You don't need to fill anything. And Python, you need to have a Python installed, but it is not it is not necessary. Python only needed if you want to build a special library, or if you want to use the split database function in the MS Frag. I will mention it later. So after everything is ready, you can go to the next tab, which is a workflow tab. And we have plenty of the workflows, preset ones for different kinds of applications here. The easiest workflow is the default one. The default workflow, they basically do the closed search using MS Frag, and then filtering the FDR using Philosopher, and that's it. There's no open search, no quantification, no DIA things. But for those for the other applications such open search, you can load the open search workflow, like click this one, then load. Then all the parameters will be set to the open search. It's almost ready to go unless you have some special requirement for your data. And then mass offset, if you want to perform mass offset search, as Alex mentioned, sometimes mass offset search is a better option. The open search, you can load this one too. After you load this one, you will see that in this mass offset box, there will be a list of the mass, which is from the common modifications. MS Vega will search all those common modifications.
Yeah, so just to open a parenthesis here. So this is probably the most important option in the program, the workflow selection, because FragPipe is basically a graphical interface that brings together all the projects that we do here, and it kind of orchestrates them in a way that the analysis can be done automatically. So instead of going tab by tab and program by program setting all the options manually, all the parameters manually, we came up with this idea of offering predefined workflows. So knowing which workflow to load is probably one of the most important steps, because by selecting the workflow there on that tab, you pretty much configure all the tools in the program. Everything gets automatically configured. You can change the parameters if you need, and you can also come up with your own workflow if you want.
Okay, so there are really a lot of workflows, and I will not go through each of them right now due to time limit. Then I will use the one we are going to show for the TMT analysis, which is the TMT10 MS3 quantifications. After I load it, so here there is a global settings for the memory and the number of the threads FragPipe will use. So you can leave it as default. If you put it as 0, which means that FragPipe will detect free memory automatically and then use it. For the number of threads, it will default to the total number of the thread your computer have -1. So my computer have 12 cores, so it will use 11 threads. So then you can load the files, add the file. Right now we use one MZML file. It supports MZML or raw file. For the TMT, you should convert the raw file to MZML, but for the other workflow, you can use the raw file. Then I set the name of the directory as experiment name. Then we go to the database tab. Database tab basically let you to specify the database, and then this is the one I have. It has.
Thanks, Sean. Sorry, we have a few questions here in chat. So some people seem to be having issues with Easy Pqp. So let me see here. So apparently after the install, Easy Pqp button doesn't work. So do they need to have Python installed?
Yes, I have needed to have Python installed. If you have Python installed, you can click this one. And in this version, FragPipe will freeze for a few seconds because it's installed Easy Pqp. And then if you want to know if it's installed or not, you can click, let me do it. Okay, it will freeze. And then, yeah, it freezes. It's due to how God is dealing with this. And then after it's done, there's no message actually. But we added the message in the next release. You should be able to see it. But if you are not sure if Easy Pqp is installed, you can open the Python command line and check. We have the GitHub tutorial I will mention later regarding how to set up the Python things, all those kind of things. I will mention later after I go through all the things.
Yeah, the Python library and Easy Pqp are not necessarily for what you're going to do now. They might be necessary for DIA analysis and large analysis in case you don't have enough resources in your computer. But for now, they're not actually necessary. So you can skip those for now. And if you're having issues, we can talk in private later, or you can open an issue on our GitHub.
Yeah, so the next question is, even putting 0, the memory ratio still have the memory ratio. That may be because you don't have enough memory. But send us your error message so we can pinpoint what exactly the reason is. But most of the time, maybe your computer does not have enough memory. Yeah, it's a support FAMES data. You don't need to convert one FAMES row into multiple MZMLs. We don't do that. You have to do that for MaxCon, but we don't need to do that for our tools. We can support films natively, even with the raw file format. Don't even need to convert to XML, but not for TMT, by the way. Yeah, it's free. For the academic usage, it's totally free. Even MS Vega is also free for the academic usage, but for the company, you need to buy the commercial license for MS Vega. But for the other tools, for the MS Vega and some other tools in the future, but for the Vega pipe itself and the philosophy itself, it's free.
Feng, maybe we should read the question as well, because for the YouTube recording, I don't think they have access to the chat. Yeah, so someone's asking if FragPipe is a free software or if it needs to buy a license. So yeah, as Feng was mentioning, pretty much all the tools are open source and free to use, with the exception of Fragger, which will depend on where you are, if you're in a company or in your research institute.
So what is Easy Pqp? Okay. Easy Pqp is a Python tool from another lab. It is used to build a spectral library. And I will mention, this is basically about this tab. And I will show when to use Easy Pqp when I mention the DIA workflow.
So Mark is asking, what is the ideal amount of memory? Well, yeah, that depends. I mean, we usually recommend starting with something around 16 gigs of RAM, but you know, some analysis. Well, we are going to show like this data set we're going to use. It's quite small, so you might not need that much. But if you have like a large fractionated wholesaleized, depending on what you're doing, you might need more. So it's kind of empirical, but we recommend starting with 16 to not have issues.
So can you send a? So another one asks, can you send again the link about the configuration? So sure. So okay, let me just go through these things. I guess it's more important. So okay, so when you go to GitHub, as Felipe showed, when you search FragPipe, this is basically the first one in the search result, and then you can go to the landing page. Click this one, landing page. You will go to our FragPipe GitHub tutorial page. It have a lot of resources here. First of all, it will have the very basic resources about the using FragPipe. When you click this one, it will tell you very basic stuff, starting from configure FragPipe, which is the configure page I just showed. And there is also some other things about all those basic stuffs here, and you can take a look at yourself. And then there are also very specific tutorial about open search, method search, and some TMT related things, label free related things, and then all those how to interpret outcomes, outputs, all those list of the building workflows. And this is a setup. This setup is a little bit different from the setup mentioned here. Actually, I'm not sure why Sarah put it here. So this setup should be the one I showed before. And you can also go to this page by click this link, this hyperlink here in the FragPipe. You click this one to go to this setup page. So yeah, this GitHub landing page has a lot of resources, and you can learn basically pretty much over every almost all basic stuff by reading all those pages. And if you still have questions, as Philip showed, you can ask a question here by creating issues. So we will normally respond very promptly. And then another one is, what does person 11 means in this case? Is it again related to memory? It is not related to memory. It is related to the number of the thread you are using. For example, in my computer, I have 12 cores in total. So FragPipe will use 11 out of 12. I leave one idle so that in case you are still needed to run something else. So when the MS Frag running, it will use 11 cores. When the Iron Quantum running, it will use 11 cores too. So this is what this 11 means. It is not related to memory, but more threads normally means more memory requirement. Okay, so this okay. So yeah, let's go back to the Tmt workflow. So after loading it, I go to database tab. So in database tab, it's where you specify the FAST file. We require the FAST file have both target and decoy sequences. If your FAST file only have target, you can click add decoy here. And then you can also add contaminations if you want. If after you click this one, it will automatically add decoys here. And then you can, after if you have a custom FAST file with target and decoy, and the decoy prefix is not the default one, you can click auto detect. It will detect what is possible prefix your FAST file have. In my case, it's still REV. So it will show you how many decoys are there. It's exactly 50%, which is what it should be. Then MS flag tab, you really don't need to change anything unless you have some special requirement, because the workflow already set it for you. And then the validation, again, you also don't need to change anything unless you have a strong reason to do that. The Ptm tab is for the open search and the glycol search, so it will not be used for TMT analysis. The MS one will also not be used for TMT analysis. And then this is where the Tmt quantification settings are. So the most critical part is this one. So you need to give or create an annotation file. Right now I have this annotation file ready. I can specify the path to it. If you don't, you can click this edit and create. So it will have, it's Tmt templates. So it will have 10 rows. And then you can give name of your samples in, so you will be easier to track all your samples rather than look at those weird numbers and characters. Then rest of the settings, you don't really need to change, except if you have reference, you will need to specify the reference and then give the name of the reference channel.
Yeah, someone actually asked about this in the previous session. So here's the thing. We normally, when you have like 2 or more plexes, it's a good idea to dedicate one of the channels to serve as a reference, or some people call it a bridge. So it's usually a pool of all samples you have in all your experiments, and then that channel can be used by Tmt integrator to during the normalization calculations. So that's why it's a good idea. If you don't have, some people sometimes don't have because of they cannot manage to lose one of the channels for a pool or a reference, for example. Tmt integrator can also create what we call a virtual channel, which is basically an extra channel that will exist only for the purpose of the program to calculate the normalization across the different plexes. Yeah, so someone is asking, how does it works with multiple Tmt plexes? Yeah, so what the program is going to do is it's going to load all PSM tables from all different Tmt plexes, and it will merge them and apply the normalization and the filtering on all the data at the same time. And then the program can output the identifications in different levels. This is what Alexei mentioned before, as the gene level, the protein level, or the site level. So basically, the PSMs are rolled up to different. They can be roll up to the gene symbol or the protein ID, for example, and all the PSM quantification is summarized at that level. So, for example, if you do at the protein level, all PSMs that map to those particular proteins are going to be used. And if you do to the gene level, for example, all the PSMs that map to those particular genes are going to be used. And at the end, as we are going to show, you're going to see different tables. And those tables will have the index that can be proteins, genes, or peptide sites, for example, as rows. And on the columns, you're going to have the Tmt labels from all the experiments. So someone is asking if we have an output. Yeah, so as Fengshui is going to finish the settings of the program, and then we're going to show how what the Tmt tables, what they look like, and we're going to go through them in details. Okay, well, let me see the other questions. So Mark is asking about the cores. Yeah, those numbers are okay, like physical core 8, 16. Yeah, that's fine. That's usually what we use. Any standard good processing server is fine. In Alex's talk, he mentioned that the final step of Tmt workflow converts back to absolute abundances. Can you explain what that means? So for the calculations, the Tmt integrator uses the reference channels. So it calculates a ratio of the abundances to the reference channel. So the final table will be a ratio value. And in one of these steps, which is optional, Tmt integrator can convert back from a ratio value to an absolute value. Is this tutorial dataset available on the GitHub? No, it's not on the GitHub, but it's if you go to the May Institute website on the program listing, right next to this session, you're going to see a link called Docs. We added the links for all the programs and to the dataset on on there, so you can find there. Can I just use peptide lab? Yes. So Fengxiao, can you click on the group by option there? Yeah, you see, so you can choose having a gene level, protein level, the peptide sequence, the Ptms, all. So if you click on all, you're just going to see all tables being printed. I use a FASTA file manually compiled with my target sequences instead of files downloaded from some databases. In this case, when I try to add decoys, nothing happens. So one of the things you should do first, if you have a custom FASTA database, and this is probably one of the most common issues we have today, the FASTA databases, they need to be properly formatted. So the headers, they need to be in a way that the programs can use them, because there are several elements in the headers that can be used by these different programs to find the protein ID or the gene ID or the description. So if you just write a custom format using your own format, it might be a problem. So what we suggest is go to the Philosopher website, the GitHub, and we have a tutorial or a description on how to properly format the FASTA headers. We normally ask people to follow the some open standards like the NCBI format or the UniProt format or the ensemble. You can have a very simple one that you can create, but you cannot add too many crazy elements on it because it might break the processing. So start by looking at what kind of format you have and check the website to see if the format is good. If it's not, you can always get in touch with us and we can help. Where do I get the appropriate FASTA sequence databases? So that, yeah, okay. So Fengxiao, can you go back to the database? Yeah, so if you don't have a database, FragPipe can also download one for you on the fly. So let's show you how to do that. So this is Saccharomyces data set. So here we have some options. Yeah, you normally click, you can just click on the Saccharomyces over there. If the organism you're working is not there, you have to look for the UniProt ID of that organism, and that's something very easy to do. So you just go to the UniProt website, you type the name of the organism you're working, and right next to the entry that will appear on the website, there will be something called UniProt ID. So you can just paste the UniProt ID over there on that field, and then FragPipe will download the database for you, and will add decoys and contaminants. FragPipe only shows Tmt 16. Does it support 18? Yes, I think this version that we are showing right now doesn't have Tmt on the listing so far, but yeah, you can do the Tmt 18 analysis. Well, in case, even though you the version that you are showing doesn't have the Tmt 18, you can still load the 16 workflow, and then when you go to the Tmt integrator tab, you can switch. You can change the number of channels on the annotation file, right? How do you add contamination? So when you download the database, if you download the database using FragPipe or Philosopher, one of the options that we just show on that small window that open is the contaminations. It allows you to add contamination. So we basically add 150 common contaminants, if I'm not mistaken. When you add FASTA, do you recommend add isoforms? In which case should I add it? That's, well, it depends, right? So it depends what you're trying to do. For a standard experiment, if you're not, basically, if you're not looking for isoforms, if you're not looking for variants, then we suggest not adding them. Isoforms and variants can complicate the analysis, especially the protein inference step. And they are usually good if you're doing explorative analysis and if you're trying to identify new variants, or if you're looking for specific variants that can be only present in certain samples. By default, if you don't know if you need it, you probably don't need it. So I would start without the isoforms unless you really have a reason for looking for them. The download button is not active on my FragPipe. What could be the reason? This is for the FASTA file download.
That's because you don't have philosopher specified. If you remove philosopher here, I guess it will be not activated. That's the reason. Please. So that's why you need to configure those three tools, at least those first two tools, to make sure that FragPipe have the tool. So make sure it can find the tool. So if I specify it, it will be able to download. Okay. So as to these questions, yes, it is the number of logical cores. Logical cores may be equal to physical cores, maybe not, depending on your CPU settings. If you have the like hyper-thread settings, it will be doubled, the number will be doubled. So this 11 is the number of logical cores minus 1.
You know, in theory, you can run any kind of analysis with one CPU core and like maybe 2 or 3 gigs of RAM. It's just going to take a lot of time.
Can you show how to add
the more you have, the better.
In the official released version, we do not support TMT18, but in the future versions, there will be TMT18. I can show it right now. This is a pre-release version. We will release this month. So there will be TMT18 here. So, okay, I guess yes. No.
Actually, that's a good point, Feng-Shou. And this serves for everyone that is watching right now and for everyone that is watching on YouTube in the future. If you see or if you notice any difference between the version you have right now and what you are showing, that's because we upgraded the programs and because we are constantly adding new features and new elements, you might see differences. So there is nothing wrong. It's just because you probably have a different version from what we're showing right now.
So someone asked in the quant tab, I think it's alphabetical quant, right? Do we want to quant level to be 2 or 3? Looks like you have a set to 3, but the data looks like MS2 data, if I understand it correctly. It has MS3. Quantification is from MS3 and the identification is from MS2 in the data I'm going to show. So for this data, it basically have MS1 and then generate MS2 for the identification. Then it uses SPS MS3 to generate the MS3 level TMT intensities. So we use MS3. If your data has MS2 as for the TMT quantifications, you need to change it to 2. Or you can load the workflow here. For example, in the workflow, we have TMT10, which means that it's TMT10 and the quantification is at the MS2 level. We also have the TMT10 MS3, so which means that the quantification is the MS3 level. So it's really depend on your data.
Yeah, one other detail, since you came back to this screen here, there are a couple of buttons that are called Save Manifest. If you go back to the workflow, yeah, we usually recommend people to saving their experiments their settings. So if you click on the save manifest, that will save the list of files that you use on your experiment. That's always a good thing, because if you want to come back later and try to remember how you did. One of the files that it's going to be also available at the end is the workflow, which contains the settings for all the parameters used. Talk about normalization. Yeah, so Alex already mentioned a little bit about the process. It doesn't go much beyond that. So it's basically, we offer two methods. The most common one that we recommend for every process is the median centering, which is a very standard approach. But the most important thing I would say that is what the filtering that happens before the median centering, because one of the key parts that or things that Tmt integrator does is to actually filter the PSMs before they get loaded. So things like overlabeling or PSMs with low purity, they all get removed by from the analysis. And also the selection of what kind of PSMs or PEPs are going to be used for the quantification. So for example, we normally start with the unique plus razor, and that's what we recommend. If you want to really get the details about the mathematics of the normalization, we can provide. Yeah, we have something on the website, but we are currently working on a draft that will explain in details what's happening. So I guess, Feng-Xiao, yeah, you can continue.
Okay, let me go back and load this workflow here.
Could you repeat how can we obtain the file in the tab of quant?
It's not obtain. It's you can click edit and create here. So it will pop up a window, and then you can edit it here. This is how you, and then you click save, and then you can specify the location where you want to save, and then you will have this annotation.txt file.
The annotation file is basically a simple text file where you're going to have in each line the TMT channels or the iTrack channels separated by a space, and then the label that you want to add. If you don't have the annotation file, nothing's going to happen. It's just at the output, you're not going to have the label names. You're going to just have the channel names. So you're going to see like 136, 137N, 138N. If you have the file, the program replaces those channels by your labels. And if you don't have the file, you can click on that create button, and FragPipe will open that small window where you can fill the blanks, and they will be used on your analysis. Is there a way to add correction factors for Tmt Quant? No, that's actually, we don't have that in place because we didn't notice any major changes for the quantification when they were being used.
Okay. So where are we now? Okay, first file. So after I talk about first file, oh, I already go to this. So we have the annotation file ready. So then we go to the run tab here, and you specify the locations like, then you click run. Then you should be able to run the whole thing. And it will take a few minutes because the data is not very big for this data set. And then this is basically the TMT workflow and TMT that we are going to show. Oh, by the way, if you want to use MS stats, you can click generate MS stats files here to let the philosopher to generate MS stats file. And then later on, you can use that file to give it to the MS stats and then run the R programs there. And then let me go through some additional workflows I guess some of you might be interested in. So let's go to the LFQ one. LFQ is label-free quantification plus a match between rounds. As Alex already showed in the presentations, we have the very fast label-free quantification tools named IronQuant, and we also have the false discovery rate control match between rounds. Everything comes with this workflow. So after you loading this workflow, you specify the files. Let me just randomly click some files as a demonstration. It's not important here. I will not really run it. So when you select it, you might need to assign those files to different experiments. So there will be 2 columns for you to assign: the experiment name and then replicated name. The experiment name can be the empty, which everything will be in the same experiment, or alphabet and underline dash, alpha numerical and underline dash. For the bioreplicated, it can only be the integers. Yes, for this, someone asked, is the LFQ MBL workflow for DDA? It is for DDA. It is for DDA for the orbit trap or orbit trap with FAMES or DDA passive. It supports actually all kinds of data I'm aware of. And you don't need to convert to XML. You can use a raw file or you can use .d from the DDA passive. So come back to the experiment. For the shortcut, you can click consecutive, so it will automatically assign each one to the experiment and 1, 2, 3, 4 bioreplicate, or by parent directory, which will all the file will be assigned to the experiment with the name of the directory, or by file name, whatever, or you set it manually. So I will click in this way. So since I've already loaded the workflow, then my first file is also ready. Then you don't need to change anything unless you have a strong reason to do that. So let us take a look at the MS1 Quant tab. This is basically the settings for the LFQ and match between rounds. Those are some basic settings, which the name explained by the name itself. And then this is the match between rounds, the RT tolerance, immobility tolerance, very straightforward. And the minimum correlations. Do we need to change the mass tolerance level? So mass tolerance is for the MF1 because it's label-free quantifications. There's always on MS1. There's no ambiguity on the levels. So match between round minimum correlations, as Alex mentioned, we correlate, given one round, we correlate to the calculated correlations of all the other rounds and then transfer the ions from the other round to the accepted rounds. So you can specify the minimum correlation so that only those rounds with highly correlations will be transferred altogether, or you can leave it at 0. And the top rounds, the HBR, MBR top rounds is also a similar function as the minimum correlation. You just control the number of the top rounds. The ions will be transferred, and then you can leave it as a very, very big number. Because we have the force discovery rate controls, so transfer the ions across all the rounds is not a problem. So here are 3 levels of the FDR. Someone asked, does match between rounds also transfer IDs between files designated with different experiment? The answer is yes. It transfers files among all those experiments, all those rounds, except unless you set something here. So it will give you a round to calculate correlations and only transfer the peptides among those with highly correlated rounds. Dimitri asked, but which PTM takes open search? Will be there, for example, modification characterizations for fast, something oxidation of proteins. I'm not quite sure if I understand this question. Maybe you can raise your hand and we can have a talk.
Are you asking about the workflows? Because you asked which PTM. Yeah, I'm not sure. Maybe you're asking about which workflow.
Yeah, just raise your hand and or contact us after this conference, after this workshop. Another one sent us an error message. This error message shows that you are not using Java 64-bit version. So that's why when FlagPipe try to use 5 GB memory, it cannot do it because you are using 32 bits version. 32 bits version only support up to 4 gigabytes. Please uninstall your Java and reinstall a 64 version. So someone sent another error message, said cannot find the purity column. Felipe, can you answer this one?
Yeah, I don't know if you're using the versions that we are using from the currently release. We can so let's go through all the way to the end with the processing, and then we can show the outputs, and then if we want, we can follow up with your problem.
Okay, someone asked where is the raw data located? I believe Felipe have already answered it, right? It's
if you mean on the web, it depends. You mean on the website or the computer. If it's on the website, you should look for the May Institute program on their website. There is a docs link right next to this session with a PDF, and in the PDF you're going to see a link for the file.
let me see if I can get it. It's dot here. So here,
the
right?
last link, yeah.
Yeah, here. You can download it from Pride. In MS1 Quant, someone asked in MS1 Quant tab for top N irons, I see default 3. Most people use a top 10 for DDA. In my experience, should we change to top 10? Yes, you can change if you believe top 10 is better than top 3. It's top 3 is experience numbers. There's no strong reason stick to top 3. There's a quick question. How do you tell if the run is done? You will know if it is done because it has all jobs done in 3 minutes, in 2 minutes. So yeah.
Well, if you have a slower computer, it might take more time, but you should see that message on the bottom over there.
Yeah. So yeah, as you can see, the job is done, and then the TMT thing is done, and I will leave it for Philip to talk about the result files later. Let me go through these workflows first. Okay. So go back to LFQ MBR. So we are here. We are left from here. So there are 3 levels of the match between run FDR controls. You can set it or use the default values. This top 1 options is for calculation of protein intensities. There are two ways to calculate protein intensities. So first way is top N approaches. Another way is the max LFQ approaches. For our experiment, max LFQ is always better than top N. But if you only have 1 or 2 experiments, then LFQ will have a lot of missing values because due to the algorithm of LFQ, max LFQ. So you might need to use top N. In the future version, if you check max LFQ, we will print both top N intensity and max LFQ intensity in the versions that we are going to release in a week. Then there are some. Then this is a tab. This is a panel about the label-based quantification. This is mostly for the phthalate delimitations or the other isotope labeling strategies. There it supports light, medium, and heavy. You can put in any labeling you want to use using this format from this tooltip shown here. In this example, it shows the phthalate, like the medium on K and R and the heavy on K and R. And then there are also advanced options, and you can check those meanings from the FragPipe tutorial web page I just showed. So after talking about this page, I think it's safe to go to the Run tab and click Run. I will not do that for now, but I will show you the result.
So let me just ask a few questions here. Okay. So yeah, Dimitri and Gabriel are asking pretty much. I think they're related. Yeah, so something that we actually forgot to mention before is that on the Pride website, you're going to find the raw files, but you need to convert them to MZML first. So if you want to do the TMT quantification for now, using, that's one of the things that is going to be required for now on FragPap, is that you need to convert the files to MZML first. And Fengsha was able to run it because he already has the files on his computers. And Lawrence, Lawrence is asking if we can label, if you can you have N15 labeling with 98, 95, 95, 99% heavy? I don't think you can, right?
We do not supporting nitrogen, heavy nitrogen or something. We only support isobaric and isotopic. And isotopic means silane or deamination, all those chemical labeling, not the metabolism labeling. Okay, so let's go to the TMT result. I have a TMT result somewhere. Not TMT, label-free quantification result. Since I just finished talking about the ARFQ workflow, so I have this small data set with 6 experiments. Each experiment has a one folder for it. It has individual result for this experiment. So this PNG file is basically the one Alex showed in the similar one as Alex showed in the slide. It's talking about the match between ROM FDR controls, the positive and the negatives. You can check if your data has a good separations, if you have some questions about your result. Then the most important files basically should be those 4 and this mstats 1 if you want to use mstats. And those 4 is combined iron level, modified peptide level, and the peptide level. Peptide level means the peptide with the modification stripped. There's no modification, only sequence. And the protein levels. Take the protein level, for example. There will be some entries like the protein IDs, entry names, genes, all those basic informations, and then go to the number of the peptides and number of the spectral count. And the most important thing is intensity here. This data is run with the version that is going to be released. So you will see the intensity of the 6 experiment, also the max LFQ intensity. The intensity is from top N, and the max LFQ is from the max LFQ. This is from the future release. But if you are using the current release, you will only see intensity, which is from max LFQ or top N based on your settings. So this is basically the command protein looks like. For the peptide and the modified peptide, it have the similar format. It's just have all the information such as the sequence, previous amino acid, next amino acid, start and end, length, charge, proteins, and affirm protein informations, and the spectral count of the peptides, and then comes to the intensity. Iron level is the same. And if you want to use mstats, there will be mstats file here. So if you want to use the reprint cryptome for the interact proteome, as Alex mentioned, you need to there will be 2 reprint files ready for you to use in the reprinter web server. You can use those 2 files, upload them to the server, and do the interactome proteomics analysis. So after talking about ARFQ, let me answer 2 questions here. Someone asked, what is the difference between top N and the max ARFQ? So it's different algorithm to calculate the protein intensities. For the top N, it basically take the top N of top N ions from this protein and then calculate the intensity, summation of the intensity, and use Z1 as the protein's intensity. For the max ARFQ, it use some more advanced algorithms, some kind of optimizations involved to calculate the max ARFQ intensity. You may need to check the literatures to know the details of the algorithms. There's 2 literatures that you can check. The first one is original max ARFQ paper from the Ulan Cox. You just search max ARFQ max quant, this paper, this MCP paper, we are talking about the original algorithms. And then we also detailed it, I mean, with more mathematical details in our ion quant paper. You can search these 2 keywords to go to ion quant paper. Here. Basically here is a calculation of protein titer use max LFQ. Here has all the mathematical details here, and you can take a look, and it will tell you how to do the calculations, basically. So someone asked, for PTM analysis, is there a place to specify variable and a statistic? Sure, it is. It's talking about specify modifications in the MS flag tab. So there will be modifications for the variable and the fixed. For the variable modifications, you need to specify site, mass, and the max occurrence, which controls the number of these PTMs in a single peptide. You also have the control of the maximum number of the modifications on the peptide. Also, the maximum combinations of all the modifications. For the site, it can be the amino acid sequence. If it's a protein terminal, you need to use these special characters as this yellow box showed here. Take a look at this yellow box, and you will know how to specify the protein N terminal, protein C terminal, peptide N terminal, peptide C terminal modifications, and amino acid site modifications. For the fixed modification, it's already easier. You just change the mass value to a certain values here. It will be set. Okay, we have talked about the LFQ and the match between rounds. Let's go to the open search. I guess some of you is interested in open search. So open search is using open search is also very easy. You just click open search and then load. That's it. Then you really don't need to change anything. And the special thing here is that the Ptm shepherd, the Ptm tab will be checked. This tab is only specified for open search and glycoproteomics. With this tool checked, it will generate summarized tables of the modifications from your open search. I will show result here. I will show result later on. And then let's show it. Okay, I have open search performed just when Alex is giving presentations. I just perform open search. So those two, starting from global something, mod summary.tsv, and global something profile.tsv is the file I mentioned that generated by these tools. These two files summarize the detailed result of the modifications from open search. For example, this profile one have a detailed information of all those modifications, like the zero modification, which means there's no modification, there's no zero data. This peak means okay, let me give some basic information about open search. So the open search basically search the PSMs with a wide isolation window or with a wide precursor windows, rather than minus plus 20 ppm. It go from minus 150 to plus 500 Dalton. So you will see a lot of delta mass in your PSM tables, and those delta mass corresponding to the modifications from open search. It's some kind of unknown modification which you don't know before you perform the search. So with this delta mass, you can generate histograms. Then those histograms will have apex. We can do the histogram picking things. We will get apex. We will get a lower boundary of a peak and upper boundary of a peak. So the most abundant apex is 0, which makes sense because most of PSM don't have any modifications. Then it will come to the methylations, those unknown modifications, and then dimethylations, formulations, carboxylations, all those things found from open search. So this file will give a summary of all the modifications or data mass from your open search. Since I only put 2 ROMs in this data set, so there's not many found. If you have a big data set, you will see a lot of interesting things coming out. Okay, then it will have the number of PSMs, those mapped mass, those names of those possible modifications, and all those RT shift, all those kind of informations listed here. I will not go through them one by one. And then another mod summary file is like a summarized modifications. from the previous file. It's just a list of the modifications, names, and the mass, and the numbers from data set, number of PSM from data set, and percentages. So the two file is kind of very useful for the open source because it will give you a basic idea of what kind of information your data have. With these files, you can cherry pick your modifications and do the more depth analysis with the following method search or the closed search or whatever. So this is the thing I want to talk about talking about the open source. Okay, let me answer some questions. For the notation rule to dedicate isolation specifically, why do you need to identify for protein terminals, but when also add? Because those have two rules. This triangle is a marker. This bracket is specifically for the protein terminals. So this means protein. If I want to it at the, for example, like methylations or the methionines at the protein terminal, I can change this. So if I use this way, it means that so the modification only happens at the protein and terminal methionine, not all protein or terminals. So that's why this triangle needed to be there. It just tell you that it only happens at the protein terminal. For example, I can use other amino acid here. So yeah, so that's why I need to use two of them, not a single one of them. Someone asked, I got 2 errors on some test files I'm trying to run. For JNA could not load Bruker library. Second, insufficient memory. So let's go through 2 of them one by one. So for the JNA things, please check the settings here. We have this paragraph here talking about if you are using Bruker's native library, you might need to install these things. We are not allowed to distribute these things in our tools because it's a Microsoft library. So please download it and install it. And then insufficient memory, even though I have plenty of memory, well, just it's the I don't know the exact reason with this information. I needed to take a look at the log file or at least the original messages and the settings you have. And also remember to install 64 bits version of Java, not 32 bits version. Someone asked, is the search result output compatible with Perseus? Yes, it is. Perseus can take any tables, TSV, CSV. You just need to specify the major columns and the intensity columns you want to use. We actually have a tutorial of using Perseus for our result. Still, in this tutorial, in this GitHub, let me see here. Importing result to Perseus. When you click here, it will show you how to import frag pipe result into Perseus. After import the result, you can do whatever you want to do with Perseus. And someone asked, can you run MBR with TMT? No, we can't. But we will support it in the future version. But right now we can't. Someone asked, is there any documentation available explaining the columns of the open source result? Yes, it is. It is in the GitHub tutorial website and should be here. And yeah, so you see those 2 files are listed here and I click it, it will tell you the columns, meaning of those columns. Yes. Someone asked, if processing multiple raw files in frag pipe in the result folder, will there be multiple TSV files or just one TSV for all raw files and then result separated into different columns for different raw files in the TSV file? If you are talking about a psm.tsv, peptide.tsv, iron.tsv, and the protein.tsv, it really depends on your experimental settings. If you, for example, let's use raw file. Just if you don't specify anything here, everything will be in one single psm.tsv and other TSV files. If you put them into two different experiments. There will be two folders. Each folder have its own separate TSV. And in the root folder, there will be a combined TSV folder, TSV files, like I show here. I have 6 experiments. Each experiment folder have their own TSV file here. And then outside, there's a combined TSV folder here. So the number of the TSV files and how the data is organized in the TSV files is really dependent on the experiment structure you are specified here. Someone asked, is there any easy way to view MSM spectrum to confirm PTM containing open source? There's an easy way to view the MS2 spectrum in the future version. Okay, it's time to bring the pre-release again. We have this open visualization window function here. Let me show you, it should work. Yeah, it will load the data, load the TMT data I'm just using, and then it will show the annotations. But the tricky part is that for the open search, the modification is not minor not, depending on your settings. Minor not specified as variable modifications. So you will not be able to see them. You might not be able to see the modifications in your sequence. So so it might be tricky to view the open search result, but for the closed search result, it should be easy.
So while you're loading there,
I think
let me just answer a few other questions here. So I see that Samantha and Silas are having issues with their program. So it might be hard for us to help you debug the issues you're having with Easy Pqp right now. So what I suggest you to do is go to our GitHub, make an account if you don't have one, and please go ahead and open an issue describing what is the problem, and someone still today will be able to help you.
Could you talk something about the DIA workflow? Yes, I will talk about it after I finish this one. Let's go through it one by one. So regarding to the view result, there will be some there are some no bugs in the visualization tools. It's specified to a certain proteins. So I cannot view the other one, but I will try to view another one.
Thanks, Shaw. I don't think we are seeing the same thing you are seeing right now.
Wait, what?
We are not seeing the visualization tool. Are you watching something there?
Yeah, that's what I'm trying to do. Okay, here it is. So let me do it again. So after you run the flag pipe, everything is done. As I showed here, after everything, it's in the future version, by the way. The current version don't have it. After everything is done, you will have this path still specified. You just click this one in the future version. It will open another tool that you can view the spectrums. It's protein tables, PSM tables, experiment tables. There are two experiments in my demonstrations. So when you click certain proteins, it will show the PSMs found from these proteins. And then when you click one of them, they will show the annotated spectrums here. But it works very, very well for the closed search. But for the open search, since the PTM localization is kind of tricky for the open search, you might not see. It might have needed some tweakings to see the good result. Yeah. Okay, so let's I finished the open workflow. So okay, go to the DIA things. For the DIA, we have two sets of the tools that can perform the DIA analysis. So first one is DIA Umpire. It is published a few years ago that can generate okay, someone asked a question related to the visualization. Let me answer this in case. So yes, it is in the future version. The current one don't have it. Okay, the DI umpire. So the DI umpire is used to generate pseudo MS2 from the DIA spectrums. With the pseudo MS2, you can search those files as a DDA data. And we recently developed the MS Flag DIA. MS Flag DIA can directly search the DIA data without pseudo generation or those kind of things. So let me show you with some toy examples. So first of all, let's load some fake data. It's not DI data, but it's fine. That's like fake data. Then let me specify the DIA SpectraLab quant workflow and load it. Then in order to enable the DIA analysis, you need to specify your data as DIA. For example, suppose all data are DIA, you click set DIA, all data will become DIA. And if some of the data is from gas phase fractionated DIA, so you can select some of them and then set them as gas phase fractionated DIA. This GPF DIA data is mostly for library generation, not for quantification. So if you specify GPF DIA, those data will not be used in the later on quantifications. Only those DIA data will be used. Or if you have a data use that you don't need to include it in the library generation, but only for quantifications, you can set it as a DIA quant. So it will not be used for the library generations. It will be used in the quant later on. Also, you can specify DDI data. For example, there are DI, DDI, gas phase fractionated DI, and DI quant data. So the DDI data will be searched in a DDI approach, and they are used to generate a spectral library together with the DI data and the gas phase fractionated DI. For the other, someone asked a question about how about SWAT DIA science. Yeah, you can use it for the SWAT DI. Use the DI for the SWAT DI. It's just SWAT DI is DI data. Yeah. So after you specify the data types, let's make it easier. Let's do the DI first. Maybe half. Okay. Then your database tab is not important. So MS Flag, there are not much you need to change for the MS Flag. It will search DIA and DDA data separately, and then go to the FDR filtering, where in this page you also don't need to change anything. Then after the FDR filtering, you will have the PSMs, the peptides passing the 1% FDR. Then also then you send the why my Easy Pqp? Okay, my Easy Pqp is still also not available. I don't know why. Anyway, so you will see this page. Why my Easy Pqp is not available? This is for the spectral library buildings. This is what the Easy Pqp is. Okay, it is available now. So yeah, as I said, if you see this unavailable, you just click this one and then it will install the Easy Pqp for you. Let me restart it. Because after install the Python library, you need to restart it. So let me use 17. Yeah, it's available now. So when this Easy Pqp is available, you should be able to enable this tab. It will basically generate a spectral library using the peptide from the DDA or DIA data MS Flag searched. Then there will be some settings. If your data have the IRT or CIRT, you can change the settings here. Then after this tab, it comes to the quantification tab using DIAM. We basically give DIAM the spectral library generated from Easy Pqp, and then let Diane to do the quantification only. So here, Diane does not do the identification. There's no spatial library prediction. There's no library generation by Diane. Diane simply do a quantifications using our spatial library. Then the good thing of using FragPipe over Diane is that you can generate a spatial library using DIA and the DDA, and also gas phase fractional DIA. So if you have different kinds of data like DIA, some DDA, and some gas phase fractional DIA, FragPipe will generate a big hybrid spatial library from all the data, and then use it as a template for the quantifications. So that's why that's the advantage of FragPipe over Diane. So after it's running, it will produce the result files. And the result file is pretty much similar to what I show here, except for some additional files from Diane quantifications. You will see it in your files. So this is about the using MS-Frag DIA to do the DIA data analysis. See another approach using the DIA umpire to do the DIA analysis. So this time, DIA umpire will be enabled. So DIA umpire will be used to generate the pseudo MS2 from the DI data, and then both DDA and DI data. The DI data means the pseudo MS2 from DI data will be used by the MS-Frag and do the database search in a DDA mode, because right now we already have the pseudo MS2 from DI data. We can perform DDA search for it. And then rest of the steps are similar to what I showed before. So the difference of this workflow and this DI umpire workflow and this DI spatial library workflow is using DI umpire or using the MS-Frag DI. From our test, MS-Frag DI give a better result than DI umpire. So let me ask some questions here. Someone asked, can the MS-Frag perform QC on spatial library? I'm not sure if I follow these questions. Can you elaborate more about performing QC on spatial library? What kind of spatial library are you talking about, and what kind of QC are you talking about? Yeah. Someone asked, what is a file type for spatial library? Can you append a pre-existing spatial library with new data, or do you have a rerun? No, you don't need. If you already have a spatial library, you can set them to quant, only for quant, no need for MS-Frag identification, no need for spatial library generations. And then go to Diane, specify your spatial library here. You can specify your spatial library here. And to the format, it's basically the format used by spatial nut and Diane. And you can run small data and then let it generate the library.tsv. The name is library.tsv, and take a look at the format. Yeah. And someone asked, does the optional spatial library option select a preview? Yes, it is. And it select a spatial library here and used for that. Yes. Okay, so I think I pretty much finished DIA workflows here, and there are some others like the label workflows, site-like workflows. I will skip them, and feel free to have a look and try. If you have any questions, just remember, go to the GitHub page. We have all the basic stuffs here. We have the building workflows, list of building workflows, and it has some basic introductions here. You feel free to take a look. If you still have some questions, please feel free to submit issues on our GitHub issue page. So, okay, Philip, can you talk about the result file, TMT result file? Now I think we only have half an hour left.
I think we have time. Yeah.
Okay. Yeah. Okay. I will let you.
Okay.
I'll stop sharing and let you to do.
Let me take over and okay. All right. So just to recapitulate a little bit, we are running a DDA Tmt MS3 workflow. Okay, so where is it here? Okay, so we are using Saccharomyces database. Feng Xiao showed how to get Fragger configure here with the variable and fixer modifications. Pretty much everything else doesn't need to be changed unless you really know that you need those parameters changed. So if you don't know what they mean, you don't need to mess with them. We did the validation using, in this case, percolator here. So if you want to use peptide profit, you can just switch here the option. The program will run in the same way, but instead of running percolator, we run peptide profit. We run protein profit to perform the protein inference and to build the protein groups. And at the end, we performed an FDR filtering using Philosopher, generating the MSX file here. We perform MS1 and MS1 quantification and the Tmt quantification using Tmt integrator. And we got results. Okay, so I believe you can see my folder here right now, right? Okay, so there will be a lot of files generated in your folder when the analysis is done. So I'm just showing here the root folder of the project that Feng Xiao just executed. So we're going to see the database file and the binary representation of database file. We don't need to work with that. So here, this is the annotation file that we use, just in case you want to look again and see how the samples were annotated. So I'm just going to go through the most important files here. The top level files, we suggest you keep them because they can be used later when you're going through the publication, because they might contain important information about the experiments you're doing. Some of those files might be necessary when you're uploading your data to Pride, for example. So like, for example, which files were used. So here is just the list of files, that's the manifest. One of the most important things that you can have here is the log file. So we have this file here. It starts with log, and it basically contains the output from every single step and every single program executed by FragPipe. Here you can see all the parameters from all the tools. And this is especially important when you are submitting issues to us via GitHub, because that's pretty much the only way we can know what kind of analysis you did, what kind of which steps you ran or what workflow you executed, and we can debug in a better way the steps to try to find the problems you're having. But in overall, even if you're not having issues, this is one of the most important output files, so you definitely want to keep those, because they will contain the information about the analysis and everything else. Even later, when you're writing your paper, you might want to come back to this file here to understand what options you use, for example. Okay, so let's go to the results file. You're going to see two separate folders. Okay, the first one will contain the name of the experiment, depending on how you configure FragPipe, and the second one is called Tmt report. Let's go into the first one. So here we have again the annotation file. We have a text representation of the FDR filtering. So here we can see how many PSMs were considered for the FDR filtering. We can see how they were broken down into these PSM, peptide, and ion layers, showing their individual FDR scoring, the number of proteins that were identified by the protein inference, and the current number of target proteins that were approved under here in this case 1% FDR scoring. The second step for the filtering, and the final numbers here, showing that for this experiment, we got about 10,000 ions, 9,000 peptides, 1,400 proteins, and 11,000 PSMs. So all this is important later if you want to understand how well the analysis performed, or if you want to keep these recorded somehow. So we just suggest you keep everything. These files that end with pepxml or pin or just in XML, these are files that were used by the programs to perform the analysis. Depending on what you're going to do later, you might want to keep them, especially if you want to submit data to Pride, for example. But you don't need to inspect them. They will not be informative unless you're doing something more detailed. Now, the most important ones are these one here that ends with a TSV on it. So basically, all the data from the experiments are broken down or separated in what we call layers, and they are filtered individually. From the PSMs, we assemble protein lists, peptide lists, and ion lists. Okay, so these tables, they look very similar to each other. Some might be more important than others. You might use just one, or you might use all. It depends on what kind of experiment you have and what you're trying to do. I'm going to show you the two most important ones that people usually use the most, especially the PSM. So let's open the PSM table. Okay, so this is basically all PSMs that pass the 1% FER filter. I'm just going to go really quickly through the file itself, showing what kind of information we report here. You don't need to take notes or anything like that. If you miss anything, don't worry. We have on the website that we showed before, that Feng shall also show it again, all the columns of every file we have on these pages explaining what they mean, what kind of information they have. Okay, so basically here we have PSMs in different rows, and we try to put as much information as possible about their identifications. So we have information about the PSMs itself, the sequences, the charge, the retention time, all the calculated and observed masses of the fragments and the peptides, sequences. We output as well all the scores that were calculated by the search engine and by the validation tools, either peptide profit or protein profit. You can also see, so here we have the MS1 intensity, the assigned and observed modification. So the difference between assigned and observed is basically if the modification was defined on the parameter file for the search engine or not. If you define the modifications as a variable or fixed, we call them assigned, because you basically assign those on the search for the search engine. And if you are doing open searches, you're going to see this column here filled with all kinds of delta masses. So we call them observed because the search engine was able to find them by just looking at their sequence. We have a question. For TMT workflow, do you have parameters to take into account co-isolation, interference, and SPS threshold? Well, one of the things we use here, and I think it might be related to what you're asking, is this purity score. So PD gives you something called an interference column, which goes from 0 to 1, like a percentage. This purity score that we have here is basically the same thing. So we look at the precursor spectra to see how much interference we have from other peaks. So you see here it goes from 0 to 1, 1 being like 100% pure, meaning that there is no co-isolation or anything else. This is one of the things that Tmt Integrator uses. I believe that the standard threshold is 0.5, so everything with a purity of 0.5 or higher is used. The other ones are discarded. Let me see another question. Is the default workflow compatible with DDA, PRM, SIM data? I think you mean the default workflow in the frag pipe. I'm not exactly sure. Maybe Feng Shao can answer you, but
yeah,
I believe
the
DDA, right? But I'm not
DDA
sure
is
about the
for
other
sure.
ones.
PIM and SIM depend on what kind of analysis do you want to perform. If you just want to perform the peptide identifications, yes. For the quantifications, it really depends on what do you want to do.
Yeah.
I mean, you because for the PIM or SIM, you can do the quantification for the MS2 level or MS1 level or together something. Yes, go to the. It really depends. Yeah, I'm talking about the default value is closed search. It's just do the identifications and yes. Default workflow even don't have the quantifications. So default workflow should work for the DDA, PIM, SIM things. But if you go to the quantifications, if you want to use MS2 quantifications for the PIM or SIM, our tool cannot do that. But if you want to use MS1 quantification, yes. It's not about a TMT.
All right,
it's not about TMT or it's not about a PTM.
All right, so yeah, we have this purity score. We also have this unique score that tells if the PSM is unique, and unique here means if the peptide sequence is mapped into different proteins, more than one protein actually. We have this session here that tells more about the identification itself. So the proteins that were mapped by those PSMs, the RDs, the entry names, the G name, and this comes back to what I mentioned before about having a good FASTA database. If you're using a FASTA database formatted with one of the common headers that we see as in UniProt or as Ensemble or NCBI, all this will be filled with information like this. Otherwise, it will be blank because the program cannot figure out what kind of information you have on the headers. And here at the very end of the file, we have the information about the TMT channels. So here are all the labels, all 10 channels and their intensities. So this is the PSM table. Okay, it's I guess pretty much the most important one that is outputted by the pipeline. We have an alternative version of the PSM, which is the MSStats CSV file, which is basically a version of the PSM table formatted in the way that is already it's ready to be consumed by MSStats. I'm not sure later if Olga or team were going to use these outputs on their tutorial, but I think they're probably going to use it. So it's basically the same PSM table, but just with a different format. So you can import directly into MSStats. And the other table here is the protein table. So this is basically the information of all the identified proteins. So here, for example, you can see like some of the contaminants that are with identifier. These are all target proteins. So the basic information about them. If you're using data from UniProt, for example, you can see their evidence level from HuPool, their description, and here some quant data. We tell how many peptides map to those proteins. We divide them by total, unique, and razor, meaning that total is just like the whole summation of all peptides. Unique is just the peptides that map uniquely to that protein. And razor is unique plus razor. So meaning that you're going to see unique peptides plus the shareable ones that got assigned exclusively to this protein. Same thing for spectral counts if you want to check the spectral counting. And the MS1 intensity as well. The list of all modifications from all peptides mapping to that protein. And here at this and very end again, the TMT labels with the intensities. Okay, so this is what we call the raw output, because there is no normalization, there is no filtering. It's just what comes out from the FDR filtering, right? So all these numbers, all these quantifications are just raw because there is no processing done on them. Now, if we come back here to the output folder, we're going to see that there is one extra folder here called Tmt report, and this is where we can find the Tmt integrated output tables. So and here again we can see genes, proteins, and peptides being quantified, and you might ask, okay, which one should I use for my downstream analysis? We use the Tmt integrated outputs when you are dealing with the Tmt or iTrack data because they are already normalized. But if you are trying to do something different, for example, if you don't want to use Tmt integrator, or if you want to use MS-stats Tmt, for example, you start with the MS-stats table, which is not normalized. Okay, another question. Sorry, I could be wrong, but I thought both DIA and PRM use MS2 for quantification. So FragPipe can do DIA quantification by DIA and Empire, but not for PRM data.
Let me answer this one. So correct and wrong. 答案 only support DIA data, and it does the MS1 and MS2 altogether for the DIA data. But since, as you can see, FragPipe using DIA to do the quantification for the DIA, and we use IronQuant to do the MS1 level quantification for DDA. So there is no place for the quantification for PIM data. That is why we cannot do the MS2 level quantification for PIM data. But if you only want to do the MS1 level quantification for PIM data, you can treat it as DDA result and give it to IronQuant.
All right, thank you. Okay, so let's take a look here real quick about over this table. So here right away you can see two set of tables. We have like abundances and ratios. So it basically it's the same data, but the quantification on the first set is the numbers are actually ratios to the references, and on the second set they're just the numbers were converted back to the absolute abundances. And we have here them MD because we use the median centering normalization. If you use another method, you're going to see a different name there. And we have gene, peptide, and protein, and gene, peptide, and protein. That's because we don't have phospho data or any kind of PTM analysis data, so we don't see the phospho sites or PTM sites, for example. So the structure of these tables are pretty much the same, and these are like expression matrices that you see, very similar to expression matrices that you see from RNA-seq analysis, for example. So let me show you what I mean by that. So this is the gene level table. We have as an index gene symbols. Okay, they are unique here. We also include here the number of PSMs and some other information about them, like the number of PSMs, the ID of the protein, the probability, and the reference channel here. And here on all the other columns, you see the normalized TMT abundances. Now, if you have, let's say that you have two TMT 10 plexes in this analysis, you'd have on this table here 20 columns with the abundances. If you are analyzing 10 TMT plexes, you're going to have like 100 columns. So all the experiments, all quantification from all the observations from all experiments are going to be summarized in this single table, right? And their labels are going to be as you pointed on the annotation file. So this is a pretty simple table. It's just this. It's just the indexes and the quantification. They are negative because of the way how we normalize. I don't recall exactly how the calculations they work right now to make them look like that. But when you observe them as if you do like a box plotting of, well, I think they're negative because I opened the ratios, right? Yeah, there you go. They're negative because of the ratios, but that's because of the adjustment to the reference channel. These channel values are normalized with the reference channel. Yeah, so these are normalized using an internal reference. And so yeah, we were looking at the ratio. That's why some of those are negative. This is the absolute values. So it's pretty much the same table here. We have the genes symbol as indexes and the quantifications in columns. So let's look at the protein here real quick. And so the protein, you have like the protein ID, it's the section number. Depending on what kind of FASTA file you have, this might be different. We also include the gene name here. I think in this column, they might not be unique depending on the settings on Tmt integrator. And here also the abundance of values. And that's it. That's basically the tables that we use. Those are the outputs we use. And as Feng Xiao mentioned, if you have more than one experiment, either Tmt or iTrack or label free, you're going to see an extra set of tables on the upper folder, upper level, which is basically the combined list of all peptides and proteins that you observed in these experiments. So I guess that's it. That's what we had to show you. If you still have questions, please take a look at our website again. Check if you can find the answer to your question there. If you have more questions about these tables and their formats, you can also look at the website. We have like description of them. If there is anything out of place or you're still confused, you can always open an issue. It doesn't need to be like a bug report or anything like that. Some people just open issues to ask questions. That's fine. And then we can get in touch and help you. Do you have anything else to show or to talk about, Feng Xiao? No? Okay. So if no one else has any other questions right now, I think we can wrap up. Yeah, okay. All right. So I don't see any other questions. So I'm going to stop the recording, stop sharing. And I don't think Olga is coming back right now. So we can just adjourn for now. And yeah, thank you for your time and thank you for staying for so long watching what we developed. And yeah, we're more than happy to help if you have any other questions in the future. All right, bye.